Good. So, good morning, everyone. It's my great pleasure to officially start the new Interdisciplinary Horizons in Psychological Research conference series. And I want to start off by thanking you all for being here today, both physically, in person, and virtually in our YouTube channel. And I want to especially thank the director of the Faculty of Psychology and Educational Sciences of the University of Coimbra, Professor Paula Paixão, that's here today. And officially start the new, I think we have feedback from the YouTube. Horizons in Psychological Research conference series. And I want to start off by thanking you all. Sorry, it's good to hear my voice. It's not. Yeah, right. And I want to especially thank the director. Anyway, so I want to thank Paula Paixão for being here today. Só tirar o som, tira mesmo o som do YouTube. Deve ser isso. And it's for support both to this conference and overall to our work in the ProAction Lab. And I want to thank all the great speakers that we have today. I'm very excited about your talks, but we'll talk about them later. I think Óscar is going to talk about them later. So this conference series is generously supported by the International Brain Research Organization, IBRO, and the European Research Council, the ERC. And in this series, we're going to have 3 conferences. This is the first one. And we're going to discuss topics that are important and perhaps transformative to the psychological sciences, and specifically the psychological sciences in Portugal, both the future of psychological sciences and perhaps importantly, the present of psychological sciences. This series came about because the departments of psychology in Portugal have long turned their backs to what's happening outside, worldwide, in terms of the psychological sciences. This is true both here in Coimbra, but elsewhere. And it's clearly visible if we look at the faculty that we have, tenured faculty that we have in our departments. So if we look at the faculties, what we see is that more than 50% of the tenured professors work in clinical psychology. More than 80%, around 80 to 90%, work in applied psychology. And then there's the rest of us, 10%, which include one statistician, which is here today, and the remaining people. And so we're talking about, I would say, no more than 10 people, or 10 professors, 9 professors, 8 professors that work in non-applied areas of psychology. And when we think about cognitive science, for instance, we're talking about 3 people. When we talk about neuroscience, we're talking about 2 people. Out of 51 or out of 60 professors, 51 professors. When we talk about animal neuroscience, which we're going to talk about today, here there's no one working on animal neuroscience, or animal science, animal psychological science, or comparative psychology, if you will. And that would be fine, except that that's in stark contrast to what happens in departments worldwide. Not only, pretty much everywhere actually, but if you look at those departments that should be an example, namely those that are ranked higher in all the rankings that we can look at, we see that we don't have this kind of distribution of applied to, or the ratio applied to basic science. This is problematic, and in fact, this percolates to other areas of science. Very recently, about 2 weeks ago, some of us, about 51 psychology researchers and professors, signed an open letter to the Foundation of Science Technology in Portugal regarding the imbalance that we saw in the evaluation panel of projects of the Foundation for Science, where we had, again, pretty much the same in the sense we had more than 50% were clinical psychologists, more than 90% were applied scientists, and then there were 3 lone researchers for the full gamut of cognitive science and basic psychology evaluation. And that also then percolated also to the amount of projects, or the number of projects that were recommended for funding on the basic sciences. One single project was recommended for funding. Now, how does this connect to our meeting today, our conference series? Well, it connects because that's really what we want to do. We want to showcase what is being done in areas that are important and, again, transformative to psychological sciences. And because the future is now, and this is one of those sentences that we always say, but it's true, the future is now. This is the right time, this is the high time for us to speak about what we could do as psychologists and what these perhaps interdisciplinary crossovers between different disciplines could help psychology in Portugal. And so that's why we have this meeting today, and what we want to do is we want to listen to these fantastic speakers, their research, be inspired by them, and at the same time think about what we should do as psychologists. Not as basic psychologists, not as applied psychologists, but as psychologists, and as a psychology department. Where should we go? And this is what I invite you to do. I invite you to take in these talks and think about how much better it would be if we had a balance, the right balance between applied and basic sciences, and how that would be important not only for psychology as a science, but also, and importantly, to the applications of psychology in the world. So, because we're relatively late and without any further ado, I'm just going to thank you all for being here again. And I know this is going to be a great day. Again, thank you very much. And I'm going to pass it to Paula Paixão. Thank you.
Good morning to all the organizers and participants in this international conference. It is indeed a great pleasure for me as Director of the Faculty of Psychology and Educational Sciences of the University of Coimbra to be here today greeting the organizers and the participants in the new Interdisciplinary Horizons in Psychological Research, the first, as Jorge said, of a series of international conferences of what will undoubtedly be a seminal event in the recent history of basic psychological research and science, because its ambition is to set the standard for what the psychological research arena and its interdisciplinary interfaces in Portugal should be for the upcoming decade. It is not an easy task, but it is certainly a very challenging and important one. In fact, to explore and expand the interface between neurosciences, cognitive science, computer sciences, and technology constitutes a mandatory mission, not only for the near or distant future, as Jorge said, but especially for the present research framework where the future is thematically being built. We are honored for having the opportunity to host such an important event. Allow me to give a special greeting to its organizers, Professor Jorge Almeida, Óscar Gonçalves, and Dr. Daniel Ribeiro. Professor Jorge Almeida has made a distinctive contribution to the research and also the teaching in cognitive neurosciences at the Faculty of Psychology and Educational Sciences at the University of Coimbra, and at the University of Coimbra since his arrival here, which has been followed by a fruitful research path that has culminated thus far with the grant that he has received from the European Research Council for the project Content Map. This path has certainly contributed to reinforce and expand the century-long tradition in experimental psychology that was initiated at the University of Coimbra and in Portugal with the creation of the first laboratory of experimental psychology in 1912. If you have some kind of curiosity about this celebration, you can see the exhibition dedicated to this event that we have in the floor above this auditorium. Professor Óscar Gonçalves has joined us more recently at the Faculty of Psychology and Educational Sciences, but he has already been innovating our approach to studying and teaching psychological neuroscience. He has a well-known and established academic career, both in the national and international arena, since he has worked in several European and American countries, namely in the United States. His vision and his projects will certainly have an impact on our research approaches, in our research approaches and paradigms. This international conference is most certainly a timely one Once it is supposed to provide us strong and useful highlights when trying to answer the relevant question, what should the psychological research be for the upcoming decade? In order to do that, the organizers were able to gather an impressive group of invited speakers who will certainly take us on an exciting ride in newly explored territories where the frontiers are constantly being redefined. We can hardly wait. This series of international conferences is hosted by the University of Coimbra, the oldest university in Portugal, and an institution for which the notions of a rich past history and future strategic planning constitute the cornerstones of today's concerns. The Faculty of Psychology and Educational Sciences addresses those same concerns, George, and we are very aware what is being discussed everywhere in the world. And we are trying to, in our mission statements, goals, and curricular organizations to provide the correct answers or useful answers that will help in this debate arena debate. This conference is co-sponsored by the IBRO, the European Research Council, and the ProAction Lab, which is working on cutting-edge research within cognitive neuroscience and psychology. It has all the ingredients to be a very successful one. I hope that you really enjoy these first international conferences in the series, and sincerely hope that it will have an impact on further research projects and ideas that will be carried on. Enjoy this day among us.
Okay, we're going to keep on moving. Thank you, Paula, again. Óscar is going to introduce the conference, the first conference of the series, and Paula, Patrícia, you can come, and Pastor, you can also come over. Obrigado. Pode sentar.
Just before introducing our speakers, allow me a few minutes to explain the reason for starting a series on new interdisciplinary horizons in psychological research with a seminar on the role of molecular and animal neuroscience in psychological research. Why to venture into this when the academic mainstream in psychology seems to be drying out more than 1 century of methodological and conceptual developments in animal mind and behavior? Why to bother when it is difficult to foresee any horizon for the establishment of new animal labs in psychology departments? And the few who are still struggling for survival seem to be undergoing a process of somehow random migration in the hope of finding a better ecological niche. Just to quote an international referee in charge of an external assessment of research centers of psychology in Portugal, why to waste time with animal models when we have computers? Let me address today 2 reasons for our session today. First, an historical one that has to do with the very own identity of psychology. Few decades after being launched as an experimental science, our founding fathers faced a methodological crisis. How to gain experimental control in studying the mind? how to be objective in approaching subjectivity. As it is often the case, this crisis led to a rebirth. John Watson, in its Behaviorist Manifest, claimed that psychology should be approached as, quoting, a purely directive experimental branch of natural sciences. And therefore, again his own words, that animal psychology should be seen as a field of study as valid as human psychology. In the next decade, psychology was going to witness an increase in methodological scope and sophistication, with inputs, among others, from ethology, evolutionary biology, or comparative psychology, contributing to a better understanding the mind and behavior across species. For the establishment of some of the most important psychological laws, we are in debt to the benchwork of animal psychology researchers. But there is still a second reason for our session today, which I refer as the interdisciplinary outreach. Allow me to ground again this on a brief historical excursion. Following up on Watson's programmatic statement, one of his contemporaries at Hopkins, Carl Lashley, decided to move what I believe is one step further and take advantage of a less complex animal model, the hydra, to start looking at the relationship between the nervous system and the conditioning mechanisms. This was going to be the beginning of a fruitful interdisciplinary relationship. Lashley ended up becoming the director of what was, at that time, the most important research lab in animal brain and behavior, the Yerkes Primate Center in Florida. A leadership that he left few years after for the neurosurgeon Carl Pribram. At Yerkes, Lashley was also the mentor of Roger Sperry, who became a Nobel laureate in 1981 for his work in split brains. Suddenly, most of the methods developed in animal psychology labs were being spread, as anticipated by Watson, across fields and being used by biologists, biochemists, physiologists, and physicians to explore the relationship between brain, mind, and behavior. Not surprising, the methodologies of our allied natural sciences were evolving dramatically as well, by increasing their resolution from the brain systems to cellular, molecular, and genetic levels. Probably the best iconic example of this work is Eric Kandel, a psychiatrist converted to neurobiology, studying the synaptic mechanisms at the cellular, molecular, and genetic level of some basic learning paradigms, long-term and short-term sensitization and habituation. He ended up being awarded a Nobel Prize in 2000 for his work. His Nobel lecture words are really enlightening. I'm quoting. "From the beginning, my purpose in translating questions about psychology of learning into the empirical language of biology was not to replace the logic of psychology." I'm repeating that. Was not to replace the logic of psychology with the logic of cellular molecular biology, but to try to join these two disciplines and contribute to a new synthesis. No wonder that for addressing the new horizons of psychological research, we have today the contribution of a panel of outstanding speakers researching on the frontiers of animal mind and behavior, molecular biology, and genetics. We believe there is a bright future ahead of us by resuming our identity as the science of mind and behavior, this time in the larger context of an interdisciplinary cross-fertilization. But in order to do that, we need to do something about it if we want to have a future. And better be fast than slow. And only then can our models claim their well-deserved awards. Thanks, and thanks for being here today, and have a great seminar. Well, while we are doing arrangements, let me introduce our first speaker today, Professor Patrícia Maciel, who's currently an associate professor at the School of Medicine, where she's a director of the Master of Science program in Health Sciences and a senior researcher at ICVS, where she develops work in the fields of neurogenetics. Starting from the gene identification in human patients, her team uses molecular biology approaches to develop diseases models using cultural cells, C. elegans, and mice models. Additionally, she is she's in charge of teaching genetics and behavior in the School of Psychology at the University of Minho, and I wish her good luck in this endeavors. She holds a Bachelor's of Science in Biochemistry and a PhD in Biomedical Science, Genetics from the University of Porto, having developed most of her experimental work at the Hôpital Necker Enfants Malades in France and the Center for Research in Neuroscience at McGill University in Canada. The title of her presentation today is Genetics of Human Neurodevelopmental Disorders, a window into the building blocks of human behavior. Thanks, Patrícia, for accepting to be with us today in this inaugural session for our series, and welcome to the University of Coimbra.
Thank you, Óscar, for that very kind presentation. Just trying to see if this works. Okay, and thank you both, Jorge and Óscar, for the invitation to be here. It's a great pleasure to be with you today and actually to attend all the rest of the conferences, which I really think the program is very, very interesting. So I hope I don't disappoint in terms of what I bring to the discussion table today. So when Óscar invited me, I thought, looking at the other contributors, I thought my contribution today could be a bit more to discuss about human approach, human genetics approach. Also, my lab also, as Óscar said, we also have animal models, but I will not focus so much on that today. So, okay, it doesn't work. I have to go here. So the idea for me today is to share with you how addressing human neurodevelopmental disorders from the human genetics perspective, how can this enlighten us in terms of our understanding of the human mind and psychology. And I can tell you already before we reach the end that, of course, we don't clarify, we don't give absolutely clear answers, but I believe that are building and actually adding up important pieces of information to this. So neurodevelopmental disorders are quite prevalent if we consider them as a whole in terms of, for instance, thinking of children will go up to 15% of children will have some kind of neurodevelopmental disorder, including attention deficit and hyperactivity disorder, developmental delay, which is defined in the beginning of life, and then can remain as intellectual disability, formerly called mental retardation, but now replaced by this designation. Specific language disorders, tic disorder, and autism spectrum disorder. Cerebral palsy can, depending on the perspective, yes or no, be included in this. And of course, many of these also have as comorbidity or seen as comorbidity epilepsy. So in a sense, it's usually typically not included, but it can be a part basically of this spectrum of presentations. Also, schizophrenia is not typically put into this label, but we recognize that there is an important neurodevelopmental component also for schizophrenia. And so the idea is that maybe if we find genes that are relevant for this type of disorders, we can understand something about the normative development and learn about the basis for human behavior. Of course, we know that these disorders have causes that are both environmental and genetic. And I think any geneticist worth its name recognizes that there is no debate between nature and nurture, right? We all know that our nature is to interact with nurture, so there's no point in having this division. We are trying to understand the genetic basis and then integrate this into the whole picture. Basically, that's the concept. So why identify, I will try to, because I also thought in the beginning, as I was preparing actually to teach these psychology students that Óscar was mentioning, that there is a bit of a dark history for genetics in every field, but I think in the psychology field specifically, and so that there may be some fear, for instance, because there are really dark moments, for instance, of eugenics and sterilization of people with intellectual deficits in the US until quite late, and in different countries. So there is a bit of a dark history. Is there a good reason why we should look for genes? I think I will present three orders of arguments. The first is more a clinical perspective. So from the clinical perspective, I think it's important for us to organize these diseases and classify them appropriately, and I think the genetic cause is part of this classification and should be included. So just for us to be talking about the same things, I think it's an important aspect. Then from the clinical perspective, it's important also for us to define genotype-phenotype correlations, and for the patient perspective, to try to be able to have some sense of where the development is going, so establish a prognosis, and so be able to predict something about what abilities will be achieved, what can be achieved with different interventions, and so on. So adequate clinical follow-up is maybe a consequence of having a proper identification of a genetic cause, for instance. Of course, then we start understanding a bit more once we identify the genes, the neurobiological processes. I will come back to that. And as you will see today in several examples, we have then the possibility of generating adequate animal models and cellular models, because we can introduce the specific changes that a human patient has into these models, and then test for instance, using both mouse, but there's really a variety of models now available, from fish to nematodes and different squid and different things. We can understand the mechanisms. And the final goal, and I think I really have this perspective, is that this type of disorders should be, and there's no reason why they will not be addressable as any other disorder. So now we think of drugs for many other diseases, and we kind of put the neurodevelopment into a place where it can't be reached by treatments. But I think that if we understand, we will have the mechanisms, we will have windows into therapeutic approaches. And so this, in my perspective, is feasible. And for some diseases, for instance, in my lab right now, we are testing drugs for one specific syndrome for which the gene was identified in 2000. The field has developed, and the companies, pharmaceutical companies, I'm actually collaborating, so it's not an academic effort, it's really pharmaceutical companies investing in testing drugs for this disorder. So it's not impossible, it's like a proof of concept for some genes which were discovered longer time ago. So from the family perspective, so this is the clinical perspective, from the family perspective, from a child who has a developmental disorder, what can be gained? I think identifying a biological basis can really relieve a lot of guilt and help the parents deal with them, because families with children with developmental disorders really have this burden of maybe I did something wrong and that's why the child is like this. So this is a very liberating, even though if it's not necessarily optimistic and good news, but it's relieving. Give a name to the problem, which is something that also helps, and allow, for instance, patients, once it has a name, we can join other people who have the same problem and do something about share strategies and so on. So this, from the family perspective, is quite relevant. And if this is a case where the basis is really clear and we can predict outcome from the genetic test, we can even allow prenatal diagnosis and actually be sure that people can have more children without having this problem, which could be very heavy for them, right? From the perspective that brings us here today, I think the point is that identifying causative genes can give us a mapping of the cellular components that are important for building up a nervous system in a way that it leads to the normal physiological behavior, so the normative human development. And that's a bit how, from the scientific perspective, we think of it. So what I will do today is talk to you a bit about what the new genetic study detection methods that we have has brought to us in terms of the perspective of the field. So I will touch not so in small details of my work, but I will give an idea of some things in which we have participated. I decided to give more a broader perspective. I will discuss with you some new concepts of risk. It's not really new, but what has emerged in terms of causative genes versus risk loci, so genes that bring risk but are not necessarily 100% defined in terms of their effect. And then I will try to show you how the field has evolved with giving you the example of the autism spectrum disorders and what have we learned in terms of the biology and of those components that I mentioned to you. So first, I will not spend any time detailing this, but just to let you know that in recent years, basically from the early 2000s on, we have been able to develop methods of genetic analysis that allow us a really large-scale analysis of our genome. So while before we would take a disease, make a hypothesis, and think maybe this gene is important for this and go check, we now are able to look basically at the whole genome without having to put any hypothesis and see what comes back. And so we have, first this was possible and with a very high resolution, as Óscar was saying. So for instance, the karyotype, which was the classical analysis that was done, the resolution is of around 5 to 10 million base pairs. So big changes or deletions, duplications, big changes are detected with these methods. Then we had some tools to go with a narrower focus, but for regions that we hypothesize are relevant, so only candidate regions. And now we started having methods in which we look at all the genome with basically 1,000 higher resolution, so we can detect much smaller things and even more smaller now. So we really have a very good way to look all over the genome, basically with these first methods that we developed. And now more recently, this what you probably have heard, next generation sequencing, which is basically a way to, the difference is that we basically break the genome in very, very tiny pieces and sequence each tiny piece at a time. And this has allowed a much cheaper and fast way to look at the whole genome, which requires a lot of computational power because it's like tearing a book in many little parts and then reading each little part and then putting it together and reconstructing the book. So that's basically the computers then do the reconstruction for us, and that's why it was possible only recently, because the power is there only recently, and we can do this basically to actually look at the whole genome, which is what's represented here. First and more available for now is the whole exome. So basically all the coding regions of the genes we can access right now for the same price as doing one gene 10 years ago. Okay, so I can just do all of them and without putting any hypothesis. And then I correlate what I find in the genome with what I have in the clinical perspective or the phenotype of the person. So we are basically now what we call the gene-first approach. And this was first, proof of concept was presented in this high-impact journal, in medical journal, the first proof of concept that this would work for diagnosis of people with intellectual disability. So basically in this article, they took 100 patients, applied this method, and saw that they could identify patients who had not had a diagnosis with the classical methods, let's say, with those that I mentioned before, karyotype and everything that people would look for. And in 16%, they could have a very sure diagnosis, and then they actually identified maybe almost 20% more that had some genes that look really promising, and with this approach, actually, many new genes have been identified afterwards. So this was just the first case, and then we would validate that those were really genes associated with intellectual disability. So we have done this kind of thing we have set up a database, let's say a cohort of Portuguese patients with intellectual disability who were not explained to date, so they had been studied with karyotype and all standard techniques, and they had no diagnosis. We took those patients and they were observed by a medical geneticist, a neuropediatrician, assessed by psychology in collaboration with actually the team of Óscar at the time, and also then analyzed with these techniques. And in these cases, so in the end, we ended up having around 325 patients plus their parents, because it's important then to exclude or to test if things are also present in parents or not, to see what is relevant for disease. And we identified alterations in deletions or duplications in 30% of the patients. Not all of them were clearly causative of disease, so around 13% had alterations that we thought, okay, we think that this is really pathogenic. But more importantly, I think, is that with this, most of them, it was heterogeneous. So we didn't have two patients with the same thing. Actually, we had maybe, I think, two patients in the whole group who had the same thing, but basically each one had a different thing. So then we go back to this construction of what does this particular change give in terms of clinical presentation? What's the phenotype, even at the psychological level? For instance, is there a specific trait that is present in everyone who has this specific genetic change? So it's a reverse syndromology, as they call it. We define the syndrome, but for that we need to go collect patients with the same change all over the world. And so this started a really large collaboration between people working in this field. We can't do this without collaborating internationally and identifying the 300 or 200 patients who have this same thing, and now let's find what is their clinical presentation. Okay, so this is really interesting, and for this, with the patients that we took from this small Portuguese group, we basically contributed to defining 6 new syndromes. With the publications, of course, these are papers with many authors, because we take people from all over the world to describe this new disease, okay, because all of them are rare. What, in terms of genes, those pieces that we found, what were the biological clues that we got? It's still quite vague. So what is appearing actually all over in terms of intellectual disability is a lot. So some proteins, for instance, cell cycle regulators, have often to do with cell proliferation, which is important in the beginning of the nervous system development, and often is associated with, for instance, microcephaly or macrocephaly, so a dysregulation of this proliferation. And other themes that have emerged is regulation of transcription, regulation of expression of the genes, transcription factors, chromatin remodeling, and of course some signaling, neurotransmitter receptors and things like that also appear, but these are new themes that we were not expecting perhaps so much. Then we did the same, for instance, now the next generation sequencing, we applied it to us the opposite way. So we took a very homogeneous group of patients with a clinical presentation similar to one syndrome that we call Rett syndrome that combines intellectual disability, motor problems, and some features of autism spectrum disorders. And so this is caused by a main gene, and this study is, but then there's other patients who have the same kind of phenotype but don't have that gene mutated. And our goal is to identify what other genes give this same type of phenotype. And it's a quite intriguing phenotype because it has regression of development at around 6 to 18 months. So it's children develop apparently normally and then regress. So it's a very peculiar, interesting, intriguing phenotype, even though quite dramatic. And so what we identified variants that we thought were explaining this disease in these patients. And then we tried to link it to the genes that, for instance, the main gene that causes this disorder is this one. And actually, I don't think it shows very well in this projection. But the new genes that are here in yellow that we identified in these patients, functionally, we could identify connections with genes that were already involved in this type of clinical presentation. So what we are trying to build here is, okay, these are rare diseases. Each one may be caused by a specific gene, but is there a common pathway that we can identify that can be, for instance, a target for treatment? Is there something that they are doing together? Is there a common function? And this is a lot of how we are thinking about it. Of course, to make our efforts look dwarf, now there are many projects going on internationally that are this multiplied by a very large number. So one paradigmatic project is this DDD project, so Deciphering Developmental Disorders project, in which they are basically doing the same in many cities of England, many places in England with clinical centers. They apply this large-scale genome analysis, correlate to the phenotype, but have huge numbers, okay? So they will have, I think, at the 10th anniversary of this project, they had tested 33,000, or over 33,000 patients. And of course, this gives them the power to detect many more things and actually conclude solidly about the things that they are seeing. And for instance, they identified 67 new genetic syndromes based on this, associated to neurodevelopment. So this is the kind of analysis that is now being done. And just to illustrate, all these individual Nature papers, each one describing a new gene, and usually with functional analysis associated with intellectual disability, with robust evidence, because they have this enormous series of patients. And so that's a bit how the field is doing it. We really need to join efforts. Overall, now we have over 1,000 genes for intellectual disability that have been identified, and bioinformatic analysis suggests that there are still more to discover. Some, of course, with different penetrance. So overall, what we had, this clinical diagnosis of autism spectrum disorder, intellectual disability. We already knew that it was a bit of a heterogeneous, broad topic, but it really is, in a way, fractioning the clinical definitions. We know that now there's many causes for each. And so this is kind of rarefying the field. So everyone has a rare disease, basically, within that group. But at the same time, what we are doing is trying to find the common topics so that we can address them together. And this part of the reverse syndromology that I mentioned, so basically describing, I think there's a really, sorry, I forgot to take out the mask. That's it's a bit hard to speak with a mask. Okay, so I think what is a really interesting field for psychologists, for instance, and as it is for clinicians, because we have the opportunity to now describe psychological phenotypes, let's say the specific traits that are associated with this genetically defined variants. So I think there's a lot of work to do that is very interesting, and so what somebody has said is that we have next generation sequencing, now we need next generation phenotyping of the patients, really good quality assessment with many different parameters, imaging, whatever we have, the best we have in the other approaches to link to what is being done at the genetics level. Another idea that has appeared from these studies is that Okay, so initially the easy part is to find the cause of those what we call the Mendelian diseases that we see segregating in the family. So that's what low-hanging fruit. We can identify that genetically easy. The common diseases that often have much more complex patterns and are multifactorial are much harder to dissect, and people had approached them from this standpoint. Because they are relatively common diseases, some of them, for instance, intellectual disability, it has not been looked like that, but let's say hyperactivity, it's relatively common. So the logic is then it must be caused by a common variant, right? So we looked at the common variants in the genome and did the genome-wide association analysis and found some loci, but each of the genetic components was really and that's why they can be common. This variance is that because they don't have a very strong impact. Otherwise, they don't segregate. They don't. If they have a very negative impact, they won't be common in the population, right? Because they will affect the possibility of you having a descendancy, and so they won't stay in the population. But the common variants so explain a really tiny portion of the risk of having, let's say, hyperactivity disorder. So we did identify some, but they really, altogether, they could explain a small part. There's still a lot to be explained still. And so between this part of the rare variants they are rare, but they have a very strong impact, and the common variants that have a small effect size, but even though together they still don't explain everything, we have this hole in the middle. And what this next generation sequencing is telling us is that there's some of this middle part is explained by variants that are rare, but have a high impact. So they are here, but they are not fully penetrant. So they won't be really causing a Mendelian disease, but they will increase the risk very much. And so, but they are quite different. So there's a lot of them. It's not, and that's why they would not be findable in these large GWAS approaches. So basically, there's really lots of tiny possibilities that can generate this outcome, or they can increase a lot the possibility of that happening. And so we are now filling this middle part of the graph. And so one of I'm just giving you one example of what this risk loci, so genetic variants, for instance, in this case, a microdeletion in a specific region of chromosome 15 that was initially described to be associated with intellectual disability and epilepsy and seizures. So second paper, basically describing the same thing. They already call it a range of neurodevelopmental phenotypes, but if you see, they're all around the neurodevelopmental delay, intellectual disability, and seizure. So it's still not very variable. But then another paper comes that shows, okay, the same variant causes autism and also psychiatric disorders. And so this is, and one thing that is happening is that it's not fully penetrant. So you can have a child that is really affected with intellectual disability and epilepsy, for instance, and then the parent has the same variant and has no phenotype at all, and it's high function. And then there's someone else in the family who has the same variant and has a slightly different presentation, for instance, autism spectrum disorder. But then when they started looking at those normal carriers of this variant, the risk, for instance, for depression was much higher, the risk for anxiety was higher, and so there was actually some association also with other psychiatric disorders. So this is one example, and then they really appeared in complex epilepsies later on. But I just a table. I realize now that you can't see anything. But for instance, for HD, so hyperactivity disorder, if you have this variant, you have the risk of having hyperactivity disorder is 36 times higher. So we have odds ratios that are 30-something, 40-something, 50-something. So this is what we were looking for, risk factors for these diseases. They're not cause of disease. They don't it's not compulsory that you will have the disease, but it really is something to take into account because it's really increasing very much the risk. And the same is for autism spectrum disorder, schizophrenia, for instance. Here you have 25 times increased. Of course, these estimates also depend then on the samples, and there's not always complete concordance, but there's a lot of these loci that are appearing that are important contributors to risk. And actually, the clinical community still cannot deal with this and is not able yet to communicate to someone who has this variant. this is not going so well. I just saw a patient, I received information from a patient who had this epilepsy patient. The parent is normal and has the same variant, and they received a report from the geneticist saying that it's a variant of unknown significance, which is something that we cannot I'm not sure what this means. This is written in the report, and this is not a variant of unknown significance. This has been seen, if there's an odds ratio of 36, I think you're quite sure to say that, okay, this is a problem, right? And even if it's 9 times or 10 times. But we were looking for this, but now that we have several of them, we are not being able to deal with it yet so well. So just I think maybe from the graph here you can see this is a study in a Finnish population of several of these microduplications, macrodeletions. In blue is the ADHD patients, and in yellow the control. So what we see is increased prevalence of these variants in controls, and often to give these odds ratios then of 20-something and so on. The resolution of the screen is not so good. So what's next then? So we find these things, and now what we want to know is what's inside this microdeletion, what is doing this, right? So what are the genes that are contributing to this? And because these sometimes are large regions, we have many genes inside, and we have to try to go for clues to identify which are the relevant ones. And in this case, for instance, animal models can be interesting. And so in this particular case, there was an animal model. This region is similar in mice, and so it was possible to generate a microdeletion in the mice. And the mice don't show a total overlap, let's say, with the human phenotype. They show increased aggressivity in behavioral tests. They show, this is the pattern of looking for a platform. So the wild-type mouse learns after some time where the platform is in a swimming pool and goes directly to that place. So it's a memory task, and the mice with this deletion have memory or learning problems. They have sensory motor changes, so they are reproducing some of the impairments. And unexpectedly, they did have not seizures, but they were resistant to seizures induced with a specific drug that we use to test epilepsy. But then when better analyzed, actually, they did not have tonic-clonic seizures, but they had something that, in terms of electrophysiology, looked like an absence seizure. So they had other types of electroactivity in the brain induced. So they were hypersensitive to this drug, but with a different presentation. So we maybe can use these models now to dissect, and then there's other models. I will go a bit faster. There's a second model analysis that was made. Again, they validate these learning aspects. I'll just try to go a bit faster. So basically, if we took the studies from the human, we have good models, and now maybe we can start to look at drugs, for instance, for this type of problem. And this is actually being done. When we tried to identify the relevant genes, there was, for instance, a candidate that is this cholinergic receptor, because this gene has been associated with ADHD also, and with Alzheimer's disease risk and schizophrenia. So it has been associated with risk for several psychiatric aspects. And also associated with sensory motor gating in humans. So there's already some promise that this could be the gene. However, so this has been put forward as the main gene. However, when we generated a specific animal deleting this gene, we did not see this phenotype. So this is a bit against that being the key gene for the region. And then there's other models being developed for other genes that are there and that apparently are reproducing some of the parts. So although here, for instance, in homozygosity, whereas the patients have only one copy with that deletion, so this is not really mimicking exactly what's in human. But we are trying to, but also raises the possibility that this could be a reason why, for instance, a patient has the disorder and the parent with the same change does not have the disorder. Maybe the child has some other variant within that region that would be recessive and not manifest, but now that he has this deletion would manifest. So maybe there's other genetic variants that contribute to the outcome, or interaction with environmental factors. And in this perspective, this gene regulates inflammation, for instance, or has been shown, the cholinergic receptor was shown to regulate inflammation. And for instance, for autism, which is one of the disorders that has increased risk with this variant, the inflammatory hypothesis during the pregnancy is one of the models that exists for autism spectrum disorder. So it was it attracted attention also that. So that does not exclude interaction with environmental aspects. Okay, and this is another gene. So another way to do this, which maybe you will hear something about today, is to take now advantage of the possibility of reprogramming human cells, taking cells, let's say, from the skin or from blood, reprogramming them to become pluripotent, so able to generate any cell type, and then program them back to being neurons. So we can have neurons in a dish obtained from cells of a patient. This is a way then to look at, so we can compare people who have deletions in this region with people who have maybe deletions in specific genes, or to cause the specific gene deletions artificially and compare the pattern of each individual gene with the pattern of the patient cells. And so this is the approach of the study that I'm showing here, is to compare single gene deletions with the patterns of activity, of gene transcription, and so on, of the patients. And so basically to identify the modules that are coming from, what part is coming from each gene within this region. And the themes that are appearing here, again, are a lot of transcriptional regulation. In this case, there's other themes appearing, like DNA repair, which is a bit unusual, but it's relevant in the patient cells and in some gene deletions. So the overall idea is that I wanted to convey is more how we are approaching this. How we can look at these regions then and then try to go now from the gene back to the phenotype. Risk mouse models having these risk regions that I mentioned have been, for instance, in this case, compared with in terms of expression patterns of genes with post-mortem brains of patients with different disorders. Here with autism spectrum disorder, schizophrenia, bipolar disorder, and major depressive disorder. And you see that some of them, so in red is the shared changes, the things that are shared between both. And you can see that there's some overlap of this, what's happening in the brain of this, and this, I think it's in the cortex here, of these mice, and what's happening in the patient brain. So this is another thing that we are trying to, and then we can see which genes are the relevant ones. So then in terms of the autism, just focusing on autism specifically, what we have been doing in terms of human genetics, we actually contributed to a project stemming from that initial intellectual disability project. For some of the patients had autism, features of autistic behaviors, and so were within this scope. And we contributed to a large scale. So autism, we know for a long time that it's quite heritable, so there's a large proportion of the variants that is explained by genetic factors. But in some monogenic forms, very rare, that have been, which probably you also hear about, which have been identified some time ago. But in large majority, we have a huge genetic heterogeneity, as I was telling you, for intellectual disability. So there's lots of different possibilities of reaching that overall outcome. And again, they were addressed in this perspective first, common disease, common variant, but now a lot of the things is coming from this more rare, or we understand that a lot of the factors are rare risk factors with high impact. Again, we have identified over 1,000 genes, so the challenge is to understand what are the biological clues that these 1,000 genes are giving us. And what's, for instance, from the copy number variations that I showed you, we have things going from the interaction basic, so with neurotransmitter receptors in this case, we have some transcription factors and transcription regulation. I think it's a big theme here. And especially there's a lot of synaptic proteins and there's a lot of gene regulation proteins. And this communication between these two things is going two ways, because we know that the genetic regulation helps build up the nervous system and also helps build up the synapse. But then the synapse, when it's stimulated, changes genetic regulation to reprogram the neuron, and that's, for instance, mechanisms of learning imply receiving a signal and then changing expression patterns in the long term. And so basically this interaction goes both ways, and that's probably not surprising that we are seeing this. How much time do I have? I'm done. Okay, so I will probably just maybe summarize here. So we participated in this large exome study of autism, 11,000 patients with autism spectrum disorders, which allowed us to identify 102 new genes that are genes that are significantly associated with autism. Really above, even though we test correcting for all possible multiple comparisons. So these are the genes that are now more clearly associated with autism, of which 30 are really truly novel genes. And when we analyze this from the expression perspective, where are these genes working? What are they doing? They are basically expressed in cerebellum and cerebral cortex. And when we look at the neurons where they are expressed in excitatory and glutamatergic neurons, so inhibitory neurons. So basically, the two, and not so much, for instance, we were looking at inflammation and microglia-related cells or other types of cells other than neurons, and it's not a major aspect in terms of expression of what we find. And so I will now move just to I guess I would just thank you. I wanted just to show the people in the lab that I summarize, I think, the idea. So in terms of the topic of what I wanted to, the biological themes, I think this new thing that appeared from the unbiased approach that we took is that this gene regulation expression is very important. The synapse is not so surprising. But as I said, I think it's interesting because that's where the nature versus nurture is occurring, because it really is places of responding to the environment. So we respond to the environment by adapting transcription and gene expression. We respond to the environment through the synapses. So that those are the two places where the genes or the major clues that the genes for autism are pointing to, which is interesting. Thank you. I'm sorry for that.
Thanks a lot, Patrícia. So very exciting findings and lots of food for thought. I have also several questions on my own, but I'll try to refrain and look at the audience if there is any question or any comment that someone would like. Rui? Is there a microphone around? No, it's better because of the transmission. So please state your name and affiliation.
Yeah.
I'll speak loudly. So I find it very interesting, and I was wondering, in terms of interaction between the common variants, if you have any way in looking into epistasis between all the variants that you mentioned when you have these large databases, if you have any way of checking that?
Yes, I think I have not done it, but people are doing that. Of course, people have also done, which I think is less interesting, that is just take them as a whole, right? But importantly, the epistasis is, and people have also addressed, and I think one of the that's one possibility that you can also use the animal models for, actually, then to validate the things that you find, and also to validate, to explore the interactions with the environment, because now you have the genetic factor defined, and you can use the animal models to test environmental impacts. But yes, definitely this kind of data requires the analysis of epistasis.
I was just wondering, because you have sometimes people with the same variant but with and without the phenotype, which suggests that there's an interaction with.
Yes, so there have been efforts even to use the sequencing data to see if they could find, but I don't think they have been very successful yet in identifying a clear genetic modifier that changes the path from, this has, to my knowledge, I don't think there is a strong answer to that, but it's been looked at, of course, because it's such a clear different outcome. Because, for instance, this case that I'm telling is a child that has an important developmental disorder, it has important epilepsy, and the father is fully functional. So that's why people tend to then devaluate. But then if you look at the numbers and you see that something increases the risk 30 times, it's like cigarette smoking increases 20 times the risk of lung cancer, and we think it's a convincing fact. But for this, I think the clinical community is having a hard time to incorporate these risk predictions.
Well, thanks a lot. Just one more question. João Peça.
I think it's something that may be in the back of the mind of several people in the audience, and maybe even some of the patients with which you interact, which is the fact that we're crossing a threshold regarding human gene editing. And so that's obviously going to be a concern going forward in the future, as this technology may become more readily available for some pathologies, as it already is in the clinic for some cases of blindness, for example. But I would like to get your take on how should the field of neurodevelopmental disorders move as a way to maybe help assess or predict if gene editing would even be interesting for these pathologies when compared with some others that may be more obvious, and if yes, where should we start looking at?
I will try to escape your risky question. No, but I think if we look at spinal muscular atrophy, a disorder that is purely motor, for which we identified the gene, and the first clinical trials were recently done, and children usually would die in the first year of life because their muscles lose strength and they are unable to breathe. And so eventually they die because they are not able to breathe. Gene therapy, even before going to the gene editing, right? Gene therapy replacing the missing gene has been able, I've seen the movies of children with those severe mutations that were sitting and starting to walk. And so because it's a motor disease, even though it's in the first year of life, for us it's clear. So I think there will be the possibility, maybe just by gene therapy, of correcting some of the defects. If we are able to reach the right regions in the brain, that's another challenge. And so I don't see why it should not be correctable as any other neurological disease in that sense. So for me, if this is not let's say a challenging ethical question. For me, the gene editing, of course, is a bit, especially if we are in clear-cut causative diseases, maybe less problematic. If we are in the risk loci, that's a bit different, right? So it's hard. You get into the gray area. That's why I escaped into the gene therapy, to be a bit safer, because when we start thinking of gene editing into risk of something, you get into trouble normally.
Well, thanks a lot. Let me just, in the transition, make a comment, Patrícia, saying that sometimes we are in this catch-22 on the collaboration between genetics and psychology, and that's what I call the problem of the disease paradigm. It's moving into the diseases rather than to moving into specific processes. And sometimes we tend to assume that some of the psychiatry disorders and psychological disorders are unified phenotypes. And one of the great contributions that I believe is still ahead of us is to go, to a certain extent, to go very reductionist and look at specific psychological processes. As the example as how you move from, to long-term potentiation and how that is associated with memory, and then from memory to fear conditioning and so on, and the role of the different genes, and using models like switch on and switch off and deletion and so on. And the collaboration between genetics and psychology is probably even more promising with these avenues.
I think just if you take these patients that are genetically defined now, these groups of patients, follow them, for instance, longitudinally, you start seeing developmental pathways. You can start actually also understand. So it doesn't need, what I'm saying is, it doesn't need to be so disease oriented. It can, it depends on how you look. Right? How you look at the animal models, how you look at the patient, because you can start looking at any aspect that you want.
Makes sense.
And you have the tool of having already the genetic definition in advance, so then you correlate with what you see. It can be any endophenotype that you want or any feature that you want.
Okay, well, thanks a lot, Patrícia, once again. Sorry that we have to move fast. Our last presenter in the morning is on the other side of the world, and we need to, to be a little bit strict on timings. And let me introduce Professor João Peça, who is currently an assistant professor of molecular and cell biology at the University of Coimbra. After graduating in biochemistry from the University of Coimbra, he completed a PhD at Duke University. Interesting that we have two graduates from Duke here today, even though coming from different fields, before moving to the MIT as a postdoc. In his research, he uses a combination of molecular genetics, optogenetics, and electrophysiological approaches in animal models and human brain organoids, with the aim of understanding how synaptic computations give rise to neuropsychiatry and developmental disorders, and uncovering how genetic and environmental elements translate into abnormal network activity. Today, we'll be presenting on neuroimmune challenges in autism and ADHD, attention deficit hyperactive disorder. So thanks, João, for accepting to share your experience and knowledge with us. And for you, of course, I'm not welcoming you to the University of Coimbra, but at least welcoming you to the Faculty of Psychology and Educational Science.
Okay, so thanks, João, for your talk, as always. It's always very, very interesting. And for our last talk of the first part of the meeting, we are going to have another guest, Lezanne Ooi. I hope I'm not butchering your name. But anyway, Lezanne is an expert on the use of organoids and cerebral organoids on Alzheimer's disease and dementia. This is a super interesting talk. In fact, in the last couple of years, some colleagues of ours, me and João and other people have been trying to get into this same question. So it's very exciting for us to have Lezanne here. Lezanne is currently a principal research fellow and group leader of the Neurodevelopment and Neurodegeneration Lab at the University of Wollongong, Australia, where she's also an associate professor. And as I said, we're very excited to have her here and to hear a lot more about how to use cerebral organoids in our quest to understand the mind, but also in our quest to understand neurodegeneration and Alzheimer's. Thank you so much, Lezanne, and without any further ado, the floor is yours. You can share your screen, and that's it. Thank you.
Thank you so much. Can you see my screen?
Yes, we can see your screen.
Fantastic. All right, let me just put in Great. Well, thank you so much to George and Oscar for inviting me to speak to you today. And also thank you everyone for being here as well. It's fantastic to be able to connect with you all the way from Australia. And you can see in the picture here, this is Wollongong. It's a beautiful city on the coast. And aside from all of the animals that try to kill you in Australia, it's a great place to be. So yeah, at some point, hopefully next year, our borders will reopen. And if you ever find yourself over this part of the world, I'd love to host some of you in Wollongong and show you around. It's a nice place to be. So I wanted to tell you a little bit about how we use stem cells and organoids to ask questions about neurobiological diseases. And my lab uses a range of different tools and techniques, and we usually find some difference that's in a disease of particular interest. And the lab focuses on motor neuron disease, Alzheimer's disease, vanishing white matter disease. We've also recently started moving into trying to understand how stress changes cells in the brain, and also brain cancer called glioblastoma. So we would look at changes that occur in human patient postmortem tissue or in mouse models, and also try and tie all of those findings together by understanding the changes in induced pluripotent stem cells. So one of the reasons that the stem cells can be really valuable is that the postmortem tissue gives us a snapshot of what is happening at the end of disease usually, but it can tell us about what's actually happening in patients, which is really, really important. The mouse models give us really valuable information that allow us to understand networks of neurons and more broadly what's happening in the brain and also what's happening in aging. But they cannot so much inform on complex disease or sporadic disease. And in the case, at least, of neurodegenerative diseases, the therapeutic results from mice haven't translated that well to humans. So we need something that can bridge the gap between these different systems. And the stem cells can fill that gap to a certain extent because they can allow us to ask questions in living cells. So we can start to ask questions about pathways, test different drugs, and really interrogate simple networks in either two-dimensional or three-dimensional cell models. But we do need to bear in mind that they are still an immature phenotype, and they are a simplified system, and that has both its advantages and disadvantages. And I thought we could talk a bit more about that. So in the talk today, I thought I'd go through a framework for how we design experiments with induced pluripotent stem cells and how we use those as a means to generate patient-derived living neural tissue, and talk a bit about some of the projects that we've done using neurons in simple 2D cultures. I'll also talk about a bit of what we did on microglia as well, and then move to more complex structures like the cortical organoids. And I thought we should also talk about some caveats and challenges and opportunities as well. So just because of time, I can only really give you a snapshot of some of the projects that have been happening in the lab. So I thought I would focus on Alzheimer's disease and also tell you a little bit about motor neuron disease as well, because there's something quite interesting in the pathologies of neurodegenerative diseases where you see these similar phenotypes popping up, and some of them are also relevant to psychiatric diseases. So some of the phenotypes that I'll talk about will be of interest to you for psychiatric disorders, because the types of phenotypes that we observe are things like oxidative stress, inflammation, and something that's common to both Alzheimer's disease and motor neuron disease, and also psychiatric disorders, is this property called neuronal hyperexcitability. So an increased excitability of neurons or neuronal networks. So in the case of Alzheimer's, when we think about the Alzheimer's brain, there's also pathologies that are very specific to Alzheimer's. So that's the amyloid plaques and the neurofibrillary tangles. Amyloid plaques are composed primarily of amyloid beta, but other proteins as well. And they have high levels of amyloid beta 42 peptide. And the neurofibrillary tangles have high levels of phosphorylated tau. And that will become important as we talk about the phenotypes later on. The other thing that's important to think about as a general concept for some of these neurological diseases is that there are disease-causing genes. In the case of Alzheimer's disease, that's presenilin 1 and presenilin 2 and amyloid precursor protein. So they cause an early onset form of Alzheimer's disease. But there's also what most people suffer from, so about 98% of the patients suffer from what is being described as a sporadic disease, which means that it is later in onset. But I think what's interesting about the word sporadic sometimes might make you think of random, but it's really not random, because there are many, many genetic contributions that give rise to an increased risk of Alzheimer's disease. And that's also becoming obvious to be the case for motor neuron disease as well, and lots of other neurological diseases as well. So in other words, if you have some of these variations, you have an increased chance of suffering from these diseases. They give you an increased vulnerability or an increased susceptibility. And the stem cells and the stem cell models can be really useful for trying to understand those specific vulnerabilities, because we can start to ask questions in very specific cell types. So the paradigm of how we would do that using induced pluripotent stem cells is that a patient would donate some of their cells. So normally we have done this using skin cells, so fibroblasts. So we take a little hole punch of the skin, chop up the tissue, culture it in a plate, the fibroblasts spread out into the culture dish, and then we can go through the process of reprogramming. And that's done by introducing pluripotency transcription factors. So transcription factors that drive the pluripotent gene expression program. And that's usually done in a combination of about 4, which almost always contains OCT4 and SOX2, KLF4, and now nanorgan Lin28. And then once you've got those stem cells, you can then differentiate those cells into almost any cell type of the body. So depending on your particular disease of interest, you might want to make different cell types. And then the next step is, once you've got those cells, you can start to ask your biological questions. So you might want to look at mRNA or protein levels. You might want to look at cell signaling pathways. So for example, you might want to look at uptake or phosphorylation or ubiquitination, so post-translational modifications. Or you might want to look at cell function, so the electrical properties. And that's something that we'll come on to a bit in a moment. So if you're going to set up these types of experiments, the first question you need to ask yourself is, which is the most relevant cell type to study? And this may be straightforward, but it may also not be as well. So in the case of motor neuron disease, one of the most obvious cell types to generate would be motor neurons, because they're the neurons that degenerate. But we may also want to consider culturing the motor neurons with the skeletal muscle, so that we're looking at skeletal, at neuromuscular junctions. But as I mentioned before, the other major point to consider is that we know that the glia have a major effect in precipitating the disease and progressing the disease. So the microglia and the astrocytes may also be important to study as well. So a simpler system is easier to study, but it may not reflect the disease. And these are the important trade-offs that you have to consider when you're first setting up the experiments. In general, I would say start simple and then start to build in complexity. So this is what it looks like when you reprogram the cells. You have these fibroblasts, which hopefully you can see here. You transfect them. We also put GFP in as well, so we can confirm transfection. And then you see these colonies start to appear in the dish, and you pick those out and then and culture them, isolate them, and expand them. And that's your stem cell clones that you then continue to work with. And they can be banked. And they have to be characterized through a kind of panel of tests to confirm that the cells are pluripotent. So pluripotency means that the cells can generate cell types from each of the 3 germ layers. So the endoderm, mesoderm, and ectoderm. And there's a pretty standard way of doing that. But basically, it's making sure that genes from those 3 germ layers can be expressed in the stem cells. And also, another really important thing to test is that the cells have a normal carrier type. So they have a karyotype that's the same as the fibroblasts from which they're derived, because you don't want to be introducing chromosomal aberrations during the process. So that's a really important step. And once you have those cells, you might, if you're looking at a particular gene mutation, you might want to reverse that mutation back to a wild type to give you an isogenic control, or you may be looking at sporadic disease, in which case you would have to compare multiple lines to multiple healthy controls. Once you've got those stem cells, then we would differentiate them. In the case of the motor neuron disease projects, if that might be into motor neurons. And in the old days, I say the old days, but I'm really talking about 5 years ago, that used to be using growth factors in small molecules to drive differentiation in a process that took about 2 to 3 months. So that's obviously quite a laborious process. And the reason that I say the old days is that the field moves really rapidly, and the newer ways to differentiate and to generate cell types is to use transcription factors to drive differentiation. And that is much quicker. So we now can go from, instead of the 2 to 3 month protocols, down to 1 to 2 to 3 weeks, which obviously is a big difference in terms of what each researcher can do in the lab. So I just wanted to give you a couple of examples of how we might utilize those cells and the kinds of questions we can ask in the lab. So one of the examples is from the motor neuron disease project, where we were looking at, specifically we wanted to ask the question about ubiquitination. So ubiquitination is a post-translational modification that's important for the degradation of proteins, amongst other things. It also has signaling roles as well. And it seems that in motor neuron disease, the ubiquitin proteasome system is severely impacted. And it's not entirely clear exactly why that is the case, or how that leads specifically to the degeneration of motor neurons. So that's a big question that people are trying to address. So one of the things that we did first was we tried to understand what it is about the whole proteome from motor neurons compared to pluripotent stem cells, like how that differs. So what is special, or what is more vulnerable in the motor neurons with regards to ubiquitination. So we looked at the entire proteome that's ubiquitinated using mass spec, so it's a ubiquitomics approach, and looked at the number of proteins that are ubiquitinated. So in motor neurons, there are considerably more proteins that are ubiquitinated than in the pluripotent stem cells. And of the proteins that are ubiquitinated, each of those proteins has a higher level of ubiquitination as well. So that's what you can see in this volcano plot. And so as we were doing that, we've obviously identified not just individual proteins, but whole pathways that are ubiquitinated and how that is affected in motor neurons and also in motor neuron disease. And one of the first things that we found was that the motor neurons have a much higher dependence on this critical enzyme called UBA1, which is the apex, the very first enzyme that's responsible for ubiquitinating proteins. And if you block that enzyme in motor neurons, it specifically reduces the viability of those motor neurons. And so then when we started to ask the same questions in disease, in cells from ALS patients versus controls, we were able to identify that there appears to be a dysfunction of UBA1, specifically in those ALS lines. So this is one of the mutations that we've been working on in ALS. So the gene is called CCNF, it's a cyclin F, but its role is within the UPS, and it seems to have this important role in specifically mediating ubiquitination. So when you have mutations in this particular gene, it is thought to be able to lead to protein aggregation. And one of the major phenotypes that we've been studying for quite a while is how or why there are changes in neuronal excitability in neurodegenerative diseases. So there's a whole load of contributing mechanisms focusing on not just the motor neurons themselves, but also GABAergic interneurons and astrocytes. And the network of those three cell types together all can have contributing effects on neuronal excitability changes in neurodegeneration. So sometimes it is really important to think about networks. And so for this particular project, we started off doing those experiments in the ALS mouse model, which is a SOD1 G93AA mouse model. It's a really well characterized mouse model, but it allows us to ask questions about aging and about networks that you can't really do in the stem cells. So the familial and sporadic ALS patients in the clinic show a neuronal hyperexcitability. So they show an increased response to a stimulus. Their neuronal pathways show an increased response to a stimulus. And that can happen, that can present even before motor symptoms appear. So familial patients that have been studied over time, that's been shown for those patients. And then after that hyperexcitability, it transitions to hypoexcitability as the symptoms progress. And in the mouse, what we were able to show is that that hypoexcitability then leads to the loss of repetitive firing. And that was driven by the potassium channels and sodium channels. So to then ask the questions, we go back into the stem cells to try and understand what specific mutations are doing to some of these channels. So I've told you that in the CCNF line, there was dysfunction of the UPS and dysfunction specifically of that enzyme UBA1. So what does it do to the ion channel function? What it does is, if you have a look here, so this is some electrophysiological traces or recordings plotted, where we've taken so this is the motor neurons from the patient. So the patient carries an S621G mutation and also one wild type allele. So it's a dominant mutation. And this is the magnitude of the sodium currents in blue here. So in the blue, it's the wild type. In the patient, when they carry this mutation, in the orange in the middle here, you can see it increases the sodium current. And the benefit of being able to use the stem cells is we can create this biallelic mutant, whereby we can introduce another mutation in the second allele. And when you do that, it further exacerbates that sodium current. So that gives us pretty good evidence that this specific mutation and that causes ALS increases the sodium currents. And it also increases potassium currents as well. So we see the same thing here, where this is the wild type. So these are the isogenic control lines where we've engineered the wild type allele back into the patient line. And this is the patient line here. So we see an increase in the potassium current that's further exacerbated in the biallelic mutant. So what about if we want to think about mimicking the brain in a more physiologically relevant manner? So let's have a look at the brain organoids. So when we compare the human brain, so the human brain's got 13 billion neurons. It's about 15 centimeters in length, front to back. The brain organoids are made from stem cells. So they're about the size of a pea and a couple millimeters across with a few millions of neurons in them. And the brain organoids themselves can be maintained for several months. So we've done experiments where we've kept them for over 9 months, and they can be cultured for that length of time. Although you do need to be aware potentially of cell death, and that is a major consideration, which we'll talk about later. So the neurons are arranged in a semi-organized way. So they show, if you do immunocytochemical staining to look at specific markers, you can see that the neurons organize themselves into layers, similar to the anatomical structure of the brain. And they also connect to each other and form active neural networks. So what that looks like when you differentiate those stem cells into organoids, you go from these flat cultures of, this is a stem cell colony, it gets converted into this structure that is termed a neural rosette. And then that is then converted into this ball-like structure called cortical neurospheres, which is kind of like an early developmental structure, and then into the organoids. And the organoids should express the markers of the cell types of the brain. So MAP2 is a neuronal marker and GFAP is an astrocyte marker, for example. So how closely can brain organoids match the human brain? So first, let's look at the ingredients. So like I said, the human brain, it's got about a neuron to glia ratio of about 1:1. The organoids, let's say they've got about 1 million neurons. We can also have in there about 1 million glia. It's also possible of those glia to think about splitting up and including other cell types as well. So most of the glia in an organoid will be the astrocytes. There will be some microglia in there. And we haven't done this, but other people have. You can now also introduce more mature oligodendrocytes and blood vessels as well. And I think this will be the future, really trying to push the boundaries of what is possible in terms of the physiological relevance of those organoids. And obviously this is relevant not just to neurodegenerative diseases, but any neurological disorder that you're interested in. So how closely do the brain organoids match the brain in terms of function? So it's safe to say at the moment that they show similar electrical patterns and connections as preterm babies, but certainly not adults yet. And as an interesting ethical debate, which is important in organoid research, at the moment, there's no evidence of consciousness yet, or most researchers believe that to be the case. They show similar patterns of signaling, but the signals are somewhat chaotic. But they do show network level events that are similar to what you could measure using an EEG in a preterm neonatal. Okay, so let's say you're going to develop your own program of research using stem cells or either neurons or organoids for your particular disease of interest. The first thing you have to do is find the cellular phenotypes. And this is absolutely critical to any disease modeling, because you need the phenotype to understand the mechanisms and the pathways, but also if you're going to test drugs, you need to have a phenotype to test the drug against. In the case of Alzheimer's disease, these are pretty well defined. We've talked about amyloid beta, we've talked about phosphorylated tau, neuroinflammation, and oxidative stress. And the other phenotype that I didn't really go into is the effect on the neurites themselves. So the fact that you have tau alterations affects the outgrowth of the neurites. And some of those phenotypes that I've just mentioned are relevant to quite a few neurological disorders, and they're very easy to measure. So in the case of the Alzheimer's disease phenotypes, just a couple of examples of what the data looks like. So you can use a Western to measure just the normal tau protein, and there's no difference in tau this is in a range of sporadic Alzheimer's lines or in the familial Alzheimer's lines. But when you look at the phosphorylated versions of tau, for example, tau that's phosphorylated at the 214 position, then you can start to see these increases in the level of those tau, phosphorylated tau versions. And what's interesting is that, so the amyloid beta phenotype, so in the familial disease, we do see an increase in the amyloid beta ratio because the familial disease is driven by those mutations in presenilin 1, presenilin 2, and amyloid precursor protein. And those mutations specifically increase amyloid beta 42. So in this disease modeling, we also see that. But we don't really see that in the sporadic disease. And we'll come back to that a bit more again later. And then also the phenotype that I just mentioned is the effect on neurite length. So we can measure neurite length as the neurons differentiate over, in this case, over 3 weeks. And in the Alzheimer's neurons, those neurites are much shorter. And the same is true of the familial disease neurites as well. The other phenotype that we often look for is what is happening functionally. So there's a few different ways to do this. I've mentioned electrophysiology so far, and you've heard about that in the previous talk as well. And another slightly easier way to measure signaling is to do what's called calcium imaging. So in these experiments, you use a fluorescent indicator that is, so it's a calcium ionophore, and it's more fluorescent when it binds to calcium. So when you when you open the channels, calcium rushes in, or when you depolarize the membrane, it opens the voltage-gated calcium channels, calcium rushes in, and you see this increase in fluorescence, which we can measure using imaging. So if we add either a high concentration of potassium, which essentially is the way that we're depolarizing the membrane, or we add something like glutamate, which then stimulates either the ion channels or the metabotropic glutamate receptors and causes those channels to open and calcium to rush in. So normally what you see is when you put those agonists on, you get this beautiful peak, which is the response, and then it closes, the channels close and it goes back down to baseline. So that's in the control lines. What we see in the Alzheimer's lines is something really quite different. So first of all, you can see there is this delayed response, and we can measure that time to peak, and that is delayed. And we can also see there are some examples where we have this aberrant signaling. So either the cells do not respond and non-responsive, or this is representative of those hyperexcitable neurons that seem to be having this aberrant signaling, this calcium signaling. So when we quantify that across the whole population of the neurons, so in the case of the controls, normally most of the neurons are responsive, and very, very few show this hyperexcitable response or a non-responsive. Whereas in the sporadic Alzheimer's lines, a significant proportion of those neurons are hyperexcitable. And that's also the case in the familial Alzheimer's lines, and an increased proportion also non-responsive as well. So we can use drugs to reverse those disease phenotypes, for example. So this is some work we did a few years ago with a drug called apigenin, and that can reduce those spontaneous calcium spikes. And when you do that, it also reduces apoptosis, so it reduces neuronal cell death. Some work that we've recently done as well is when we use a drug called alpha tocopherol, which is a lipid antioxidant. So this is a cellular antioxidant, and this is a lipid antioxidant. And alpha tocopherol not only can reduce the aberrant signaling, calcium signaling, so reduce the hyperexcitability of those neurons, but it also seems to be able to repair those issues with the reductions in neurite length by increasing neurite length in the Alzheimer's lines as well. And I briefly just wanted to mention the microglia as well, because this is the point that I was making. When you start to ask questions about different cell types, different cell types have different phenotypes. So in the case of the microglia from the Alzheimer's lines, the same lines where I've showed you the data on, say, calcium signaling or neurite length, you can then use to study different cell types. So in the case of the microglia, it's a different story, because so these are the lines that carry the ApoE4 mutation. So this is the sporadic mutation. And the ApoE4 microglia show a very, very different phenotype to the familial Alzheimer's disease lines. So the lines that have mutations in either presenilin-1 or amyloid precursor protein. Specifically, they cause a decrease in phagocytosis and a decrease in metabolism and and migration, and an increase in cytokine release, which is very different to what we found in the familial lines. So the kinds of experiments that you can do is looking at the internalization of either fluorescent particles or of the amyloid beta itself. And the microglia that carry an ApoE4 mutation have this reduced ability to internalize or phagocytose these fluorescent molecules or A beta itself. So this is consistent with the idea that what we've shown, I mean, and this is from the whole field as well as our lab, the amyloid, so the familial Alzheimer's neurons increase the production of A beta, but the ApoE4, the sporadic, the late onset microglia are reducing the clearance of A beta, and that's quite a different mechanism. And this work would be supportive of that idea. So finally, if we move on to what happens with the microelectrode arrays. So this is what the microelectrode, this is one of the ways that you can measure the function of the organoids. So this is what the microelectrode array looks like. It's a well, it's got the organoid sitting in the middle there, surrounded by the media. And the organoid is sitting on top of this array of electrodes that measure the electrical activity of that organoid. And you'll see in the traces as similar to what you saw in the previous talk is spikes that you can then measure the different properties of. So those spikes refer to extracellular action potentials. So you can either filter the data and look at the local field potentials or look at the extracellular action potentials and try and understand that data, because that data really tells us how the neurons are communicating together as a network. So it's quite different to your standard electrophysiology patch clamp, which really just looks at one neuron at a time. This is now starting to ask questions about how neurons communicate with one another. And that is much closer to what we would be able to do looking at, a brain. So what I think, this is the last sort of data slide that I wanted to mention, what I think the future is for the field in general, in not just neurodegenerative diseases, but in neurological diseases, and trying to understand networks of neurons. And so how we've started to do that is to take those recordings and to use machine learning to try and make sense of what that data means. So it takes iterative models to try and make sense of the data. And we can then plot that information as it changes over time. So we start to see a trajectory of how those changes happen over time and how they change in response to drugs as well. And so this may be able to lead us to some kind of personalized medicine approach, because we can take organoids from individuals and use them almost like an avatar. So if a drug works against a particular phenotype in a particular person's cells and a particular person's organoids, it may have the potential to work on that person. And so just one example of this is one drug that we tried on 2 different cell lines, and it has the opposite effect. And that can be clearly identified using machine learning in this particular way. So a few discussion points. There's caveats and challenges, like with any science. So we're still in a place where cell maturation is limited and there are high levels of cellular stress as a result of the culture. But this may actually be beneficial for disease modeling, because that cellular stress that we impose onto those cells or organoids over time is similar to the stress that they undergo with aging. So it could be a benefit potentially. Nevertheless, future work, I think, needs to focus on progressing these model systems better so we can have a better and more physiologically relevant understanding of the spatiotemporal dynamics that happen during differentiation. So trying to make it more relevant to what we would see in vivo. But there are opportunities. So stem cells and organoid technology can allow us to ask some really fundamental questions about pathology and genetic risk in a humanized system. And I think the key point, I've enjoyed all the talks that I've heard so far in the discussion as well, And I think one of the benefits that you have from working in the institute where you are is it's so important for lab scientists and clinicians to work closely together, because those technological advances that happen in the lab need to be guided by clinical insights and the most pressing health needs. So we've talked so far about how we create the induced pluripotent stem cells as a means to generate living neural tissue in the lab. The questions that you want to ask yourself are about complexity. So do you want simple cultures that you can understand pathways and biological processes like ubiquitination, or multiple cell types where you can start to ask questions about networks and function? And when you look at those disease-relevant phenotypes, you want to be thinking about what is relevant to the disease. So in Alzheimer's, for example, we talked about the characteristic hallmarks and looked at ways to measure the hyperexcitability of neurons. And then to be able to test potential therapeutics, you need those phenotypes. And it just remains for me to thank everyone in my lab who just work incredibly hard to make this happen, and all of the many collaborators and clinicians also that we wouldn't be able to do this work without as well. And of course, the funding body that provide the money. And thanks very much for listening.
Thank you very much. Okay, thank you, Lezanne. It was a great talk. I'm going to ask if there's any question in the audience for you. I'm going to ask one myself, perhaps. Well, one of the issues is that we're very close to lunchtime here, and you've passed your dinner time over there, so it's hard to ask questions when you're in these situations. Anyways, I was very excited with the microelectrode array. As I said, we have a vested interest in organoids, cerebral organoids, and actually aging. And this seems to be a great opportunity for looking at, as you said, for looking at network properties and connectivity and as such. And the first question is, I don't think you showed that, but are there differences, immediate differences between controls and the two types of AD patients that you looked at? That's number one. And then whether, because it feels like if there are, even if there aren't, but if there are, this is an avenue for understanding how this correlates or how this matches with other types of measures that we get from actual in vivo patients and subjects.
Yeah, well, it's a great question. There are differences between disease and control, but there's also line-to-line difference as well. And I think that is one of the challenges of working with stem cells, is that we have to make sure that we do so many lines to make sure that the effects that we see are significant. So it's probably easier to start off by asking about familial lines, because you've got the isogenic control and you can be more certain, never completely certain, but more certain that the difference is due to that mutation. And if we think about in the MND context, what's quite interesting is that the excitability that you see in neurons, in the motor neurons, so first of all, that starts out as hyperexcitability. If you take a disease mutation causing astrocyte and put that with the motor neurons, whether they're healthy or not, it flips the hyperexcitability to hypoexcitability. So it tells us that there is a significant contribution of the astrocytes and the glia, and understanding that time frame is probably one of the really important parts. So I think one of the benefits of the MEAs is that you can start to track over time. So what happens to the excitability changes over time? And I think that's the really critical point, because up until now, I think taking a snapshot at a particular time point and hoping for the best probably doesn't give us the full picture, and so what we've started to do, I didn't show the data, but we started to do some experiments where we started on day 21 and then measured every day for 9 weeks. And you start to see those significant changes later in the maturation period. So I guess that's the major piece of advice, is that you really want to be understanding what's happening over time, and then to make sense of that data, because you get such a massive amount of data, and that's another challenge. You know, you really need to be investing in either some kind of machine learning or some way to make sense of that massive information.
Thank you. You have a question here in the audience, perhaps two questions, but let's start with João Peça. Embaixo. Embaixo mesmo. Carrega ao lado. Isso. Okay, he's going to ask you a question.
Hi, congratulations on the talk. Fantastic work. My question is regarding the microglia. For me, it wasn't clear if you see spontaneous appearance of microglia in your organoids. There have been some reports in the literature where people do see this, or if you see the need, for example, to differentiate microglia from the original iPSCs and then see them on top of the organoids. Can you discuss this technical detail a little bit further? Thank you.
Yeah, that's a fantastic question, and absolutely spot on. That's right. So you do see the appearance of some at least early progenitors, but not many within the organoids, and that's consistent with other people that have published similar things in the literature. So if you want to ask questions about the role of microglia, I completely agree. It's necessary to seed more microglia into those organoids. And I think that's, in a way, that's a good thing, because you can then control the population of microglia that you're putting in as well, which is potentially important. But yeah, it's an important technical point.
Thank you.
We have another question by Óscar Gonçalves.
Hello, I'm Óscar. Thanks a lot for your presentation. This is really fascinating research. Let me ask you, just to speculate a little bit, building on something that you briefly mentioned, the issue of consciousness on organoids. And there was a recent debate, I believe, on Neuron, on islands of awareness, and if we can get signatures of consciousness in cell models, isolated models such as organoids. And I'd like if you could speculate a little bit about the possibilities of using microarray technology as searching for markers of consciousness in organoids.
Yeah, I mean, it is a fascinating topic, isn't it? And I think part of the problem is that we don't really have a good definition of what consciousness is in a person, in an adult, or how it switches between what happens from a neonate to during development as well. So part of the issue is that we probably don't have a good signature for that anyway. At the moment, like I said, it looks like there's no consciousness, at least from what we can see in the signals. It doesn't look to be the case. But this idea of pockets or islands, I think, so, I mean, we're not using high density microelectrode arrays, but you can get incredibly high density microelectrode arrays now. So, 9,000 electrodes or upwards. And I think, this kind of technology is moving forwards really rapidly, like not just the generation of the cells, but also how we analyze them. And as we get better at that and develop better systems for analysis, I think you could be right that we might find areas that do display those characteristics, which will lead to significant ethical debates. I mean, it already is leading to ethical debates, and it's an important question for sure, because we don't want to be working on conscious organoids.
Thank you.
Okay, thank you very much. I think there's no other questions. Lezanne, thank you so much for your talk. It was great, and we'll certainly chat later. This is as you said, very enticing, and for sure we're gonna bother you in the future.
Sounds good to me.
Great, thank you so much, and yeah, please welcome.
Thank you. Thanks.
So we're gonna have our lunch break. Again, I'm sorry for the half an hour. Well, actually, half an hour delay, but you know, it's worth it. The more you wait for a good thing, the better, I guess. I will see you guys back at 2 sharp, if possible, so that we don't go back at this later. And thank you very much for being here. Thank you.
And our first speaker will be Professor Armando Machado, who's currently a full professor of psychology at the University of Minho. Pardon me, it's slip of tongue. I was going to say, after being, for more than 2 decades, a full professor of psychology at the University of Minho, where we were colleagues, where, in fact, he launched what I believe was the first lab in Portugal in animal learning and behavior. Armando graduated in psychology from the University of Lisbon and completed a PhD at Duke University, another alumni from Duke, before joining Indiana University, first as an assistant and later as an associate professor. He was the president of the American Society of the Quantitative Analysis of Behavior, and the founder and the first president of the Portuguese Association of Experimental Psychology. His research focuses on temporal learning, numerical discrimination, choice, and decision-making in animals. In most of his studies, he contrasts data with predictions of simple mathematical models of behavior. Today, he will be presenting on the behavioristic approach, a century in retrospect and prospect. Thanks, Armando, for accepting to contribute to our inaugural session, and welcome to our university. The stage is yours.
Thank you, Oscar. Thank you, George, for inviting me. It's great to be here, and I hope that this event, a seminal event, will be pregnant of consequences, positive consequences for psychology in Portugal also. I will not be talking about my research today. I'm going to tell you a story that spans about 100 years and that reveals a paradox or an irony. I'll let you decide which. In 1913, John Watson, Oscar facilitated my work, gave voice to a set of ideas that upended the then dominant approaches in psychology, structuralism and functionalism. Instead of consciousness and introspective mental states, Watson argued that behavior should be the proper datum for psychology. Behavioristic psychology should follow the lead of the hard sciences by sticking to measurable things. It should learn from biology and look to animals as well as humans for its principles. Watson intended that behavior should be studied not as a cue to the mind or even the nervous system, but for its own sake. By behavior, he meant the activities of the whole organism in context. To do good science would require controlling the organism's history, environment, both past and present, and the consequences of its actions, while measuring all of them objectively. The hope was that, given an adequate set of experimental results, laws and principles would be derived inductively and then tested following the scientific trajectory already so successful in chemistry, biology, and other natural sciences. Proper controlled experiments, especially if they involve substantial rewards and punishments, are difficult with humans but possible with animals. Hence, the study of animal behavior has always been integral to the behavioristic approach. Behaviorism was a direct outgrowth of animal learning studies during the last decades of the 19th and the first decades of the 20th century. 2 directions became highly influential. The research of Russian physiologist Ivan Pavlov, that João mentioned this morning, and on conditioned reflexes with dogs, later called Pavlovian or classical conditioning, and American psychologist Edward Lee Thorndike's studies of trial and error learning with cats, later called instrumental or operant conditioning. What followed Pavlov, Watson, and Thorndike was laboratory animal research that greatly expanded the range of our knowledge of learning and pointed to novel applications. After Watson left Johns Hopkins in 1920 and took a career in advertising, you know there was a scandal there, laboratory work on the behaviorist tradition was carried on by the schools of the neo-behaviorists Clark Hull at Yale and Edward Chase Tolman at Berkeley. The main experimental method involved maze learning with rats. In rats, Hull's group tended to focus on simple habits. Tolman devised novel mazes and emphasized the idea of cognitive maps. Hull and his disciple Kenneth Spence at Iowa also sought out mathematical laws for learned behavior. A third school, which became the most influential, developed after 1938 from the work of radical behaviorist B.F. Skinner at Harvard. Most of Skinner's empirical research dealt with operant conditioning, which he likened to a power, the power of an animal to operate on the environment, change it, and in turn be changed by it. Animals and people are spontaneously active. Their actions have consequences. These consequences strengthen or weaken the acts that produce them. Later, Skinner would conceive of operant conditioning as the selection of behavior by its consequences, much as Darwin conceived of evolution by means of natural selection. Skinner contributed immensely to psychology, with technical innovations and refinements, the cumulative record, the Skinner box, empirical discoveries, especially reinforcement schedules, and shaping, as someone put it, discovered in a day of great illumination. Theoretical interpretations of human public and private behavior, epistemological writings on method and theory, philosophical articulations and defenses of his behavioristic approach, and proposals to apply the findings of the experimental analysis of behavior to all aspects of human behavior, including the treatment of mental health problems, the education of children, language, and the design of a culture. The study of learning in animals has led to numerous applications. After Watson and Rayner demonstrated in 1920 the acquisition of a phobia through classical conditioning in an 11-month-old boy, and 4 years later, Mary Cover-Jones demonstrated in another infant the undoing of fear conditioning, Joseph Wolpe proposed systematic desensitization in the 50s as a technique to eliminate a variety of phobias. Roughly at the same time, other psychologists started to use operant conditioning principles to help people in a variety of ways. To teach a child with autism to wear prescription glasses, to stop ruminative vomiting in a child with an intellectual disability, or to teach nurses to modify patient behavior in psychiatric wards, for example. Thus, behavior therapy and behavior modification were born, the former owing more to classical conditioning, the latter to operant conditioning. During the 70s and 80s, as behavior modification was relabeled applied behavior analysis, other researchers proposed to merge cognitive interventions aimed to change dysfunctional thoughts with behavior interventions aimed to change response patterns, a hybrid approach known as cognitive behavior modification or cognitive behavior therapy. During the last 3 decades of the 20th century, as behaviorism started to lose its prominence in psychology, a third wave of behavioral researchers became influential, conducting research on new topics, choice, self-control, concept learning, the interactions between learning and species-typical behavior, advancing quantitative models of behavior and learning, and developing research at the intersection of different disciplines. Behavioral economics, psychopharmacology are 2 examples. In addition to the methodological, purposive, and radical behaviorisms of Watson, Tolman, and Skinner, respectively, new directions for the philosophy of behaviorism were followed, most prominently teleological behaviorism by Howard Drucklin, theoretical behaviorism by John Staden, and biological behaviorism by the late Bill Timberlake. Despite their differences, the various behaviorisms share a family resemblance. By conceiving of behavior as the proper subject matter of psychology, behaviorists have always accorded fundamental importance to the environment. Behavior cannot be understood apart from the environment in which it occurs or has occurred, so much so that this idea may be the central tenet of behaviorism. I paraphrase Dobzhansky. Nothing in behavior makes sense except in the light of the environment, both past and present. The subject matter of psychology is the environment-behavior interactions of animals and people, and how these interactions affect subsequent behavior. The study of interaction yields functional relations between behavior and environment, behavioral analysis. The study of the cumulative effects of recurrent interactions is the study of the organism's history, behavioral synthesis. Behavioristic research has studied intensely three primitive learning processes: habituation, classical, and operant conditioning. I will review briefly the last two. Classical conditioning. When a neutral stimulus is followed by a positively or negatively valued stimulus, such as a ringing bell announces food, or a cracking twig announces a predator, another elementary form of learning takes place, classical conditioning. The organism learns to use the neutral stimulus to anticipate the next one, whether it will be good or bad, where and when will it occur, how strong will it be. In the first systematic study of the process by Pavlov, a hungry dog heard a buzzer, the neutral stimulus, and as you know, a few seconds later, the dog received a piece of meat, a biologically significant and positively valued stimulus. After a relatively long interval, and this is not a minor detail, the buzzer food sequence repeated. After a few buzzer food pairings, the dog salivated upon hearing the buzzer. Classical conditioning is involved in the acquisition of emotional responses in humans. A child bitten by a dog or simply exposed to a barking dog may fear the animal or dogs in general. Fear may also be acquired simply by observing another person showing intense fear in the presence of, say, an ancient enemy, a snake. Using a variety of laboratory preparations, behavioral psychologists have examined the factors that determine whether, how well, and how fast a CSUS association is learned or unlearned. Okay? The kinds of associations, excitatory, inhibitory, that can be learned easily, with some difficulty, or hardly if at all. The form and strength of the conditional response, or the effects of learning on other stimuli. They have also developed theories and models to explain what is learned in classical conditioning and to predict the effect of more complex stimulus arrangements. These evolving theories have inspired applications of classical conditioning to socially relevant problems. An offspring of these studies is the study of contingency judgment in humans, which examines how probabilistic relations between stimuli affect our judgment of their association. Is the ingestion of a particular food associated with an allergy, for example? Another offspring is the study of evaluative conditioning, inspired in second and higher order conditioning studies. Here, they examine how we learn to like or dislike objects. Pairing an abstract geometric drawing with an attractive picture or a nice word, for example, honest, tends to change the drawing's valence from neutral to positive. Pairing it with a threatening or disgusting picture or with an unpleasant word, such as arrogant, tends to change its valence from neutral to negative. Evaluative conditioning is used abundantly in advertising. Once an association is learned, how can it be unlearned? If the buzzer is presented without food, the dog will salivate less and less to the buzzer, and eventually will stop salivating to it. This extinction procedure immediately suggests a way to treat non-adaptive emotional responses elicited by specific stimuli, expose the subject to the phobic stimulus, and ensure that no aversive event occurs during the exposure. Extinction-based exposure techniques are key ingredients of the most effective psychotherapies for anxiety disorders. But extinction of the CSUS association does not seem to be the simple undoing or reversing of the acquisition of that association. Part of the evidence is that the effects of extinction are often restricted to the extinction context. The buzzer that no longer elicits salivation in the extinction context may readily elicit salivation in a new context. More generally, the phenomena of renewal, reinstatement, and spontaneous recovery suggest, but the issue is far from settled, that extinction may involve the learning of inhibitory and context-dependent associations. Be that as it may be, the study of extinction is helping applied psychologists understand the relapse of emotional reactions after their successful treatment and how to prevent them. As Vervilliet and collaborators put it, learning theory is a continuous source of inspiration for new developments in the field of fear extinction and its return. Let us now turn to operant conditioning and say a few things about it. The way that most higher animals learned. In the prototypical experiment, a hungry rat is reinforced with food for pressing a lever. Because of its consequences, lever pressing occurs at a higher rate. If afterwards lever pressing is no longer reinforced, its rate decreases typically. This extinction process is often accompanied by emotional responses, biting the lever, for example. Between the extremes of continuous reinforcement and extinction lies partial reinforcement, with only some responses reinforced. The rule specifying which response will be reinforced defines a reinforcement schedule. Much operant conditioning research has been devoted to the effects of schedules on the rates and probabilities of responding, the distribution of responses across time, and the resistance of responses to extinction. Here's a variable ratio in action. Yeah, that's my mom calling me, but it's okay, it's only the second time. A free reinforcement, you were supposed to laugh. It must be the mask. A free reinforcer may tell the organism it is in a food situation, so it may emit food-related behaviors. But if one of those is reinforced, the animal's repertoire is unlikely to increase any further. How do animals and people learn novel and sometimes complex and creative behaviors? The usual method is shaping, the differential reinforcement of successive approximations to a target behavior. In each stage, a behavior closer to the target response than in the previous stage is reinforced, while other behaviors are not. The principle is well illustrated in another cartoon. Okay, Godfrey, run out for a pass. Blop. I'm improving. At least that time I touched the ball. Okay. Shaping may occur naturally. A case in point involves female cowbird reaction, the reinforcer, to young male cowbird singing. Wing stroking by females correlated with faster rate of song development in males. It was also related to differences in song potency, the capacity of the song to attract the female. Non-vocal shaping may be like the processes that influence phonological development in human infants, according to King and Goldstein. Shaping is common in everyday human life, as children learn to dress, swim, write the alphabet, or read, for example. It has been used extensively in applied settings to teach rudimentary language skills to autistic children, appropriate behavior to severely psychotic patients, and complex performances to professional athletes. Shaping is also used to train cooperative behaviors in zoo animals. Operant conditioning stresses behavior outcome relations, but these relations typically hold only under specific conditions. Packing a key in a Skinner box produces food when the key is red, but not when it's green. The result of these contingencies is typically the acquisition of a discrimination. The pigeon pecks at the red, but not the green key. A stimulus in the presence of which a response is likely to be reinforced is a discriminative stimulus, or SD. We need some jargon here. The fox is likely to obtain a meal by chasing the rabbit, the SD, but not by chasing the hound. The child is likely to receive a smile by saying daddy in front of her father, the SD, but not to a stranger. The fox and the child have learned the stimulus discrimination to respond differently in the presence of different stimuli. Of course, the fox chases not just a single rabbit, but a class of rabbits, white, gray, and black. Chasing shows stimulus generalization. By varying the SD, the response, the reinforcement schedule, and a host of other variables, behavioral psychologists had found many empirical regularities concerning the acquisition and maintenance of stimulus control. How organisms attend and respond to selective features of their environment, how they generalize their experience, and how they learn complex discriminations and discriminative repertoires. Their techniques and findings have been used to investigate a broad range of issues, ranging from animal psychophysics, do birds experience visual illusions, to problems of applied educational, organizational, and clinical psychology. For example, how to decrease off-task behaviors in the classroom, how to train complex skills in a factory, or how to treat self-injurious behavior. An SD often becomes a conditioned reinforcer, a stimulus that can be used to strengthen behavior. Chains of responses may thus be learned, with one and the same stimulus serving as the reinforcer of the response that precedes it, and as the discriminative stimulus, the cue for the response that follows it. The actual causation is much more complex than this. The reinforcing properties of a stimulus weakens the further back in time or in the chain it is from the primary reinforcer. Chaining has been used in many applied settings. Recently, with a clicker, click, click, in the role of a conditioned stimulus, chaining was used successfully to teach complex procedural surgical skills, tying knots, making low-angle drilling holes to medical students. So the answer to the question there is yes, in the title. The initial link of a chain announces a delayed reinforcer at the end of the chain. The teacher instructs the student to solve the first 5 homework problems before going to recess. Whether the child attends to the initial link, stimulus, the instruction, and whether she continues to respond throughout the delay as other stimuli occur may depend on the child's delay of reinforcement gradient. That is, on how rapidly reinforcement value V decreases with delay D. Many studies have yielded results consistent with the following function, okay, where V sub 0 is the value of the reinforcer without a delay, and K is a delay discounting parameter. A steep gradient, for example, curve 2 here, large K, may mean that the stimuli, we have 2 stimuli here, C and D, may mean a steep gradient, this curve, may mean that the stimuli at the beginning of the chain may not reinforce adequately the behavior of attending to them, and the child's attention will wander off task. Moreover, steep gradients may also select rapid responding over slower responding, okay? Because only when behavior occurs rapidly will the effect of the final reinforcer concentrate on the behavior and affect it. In addition to attention problems, steep gradients may engender hyperactive behavior, as in children with attention deficit hyperactivity disorder. How am I doing on concerning time? 10 minutes? Yeah, more or less. Okay. I was going to skip choice and matching and self-control, but I'll go for it. Don't be impulsive, Armando. Reinforcers may vary along multiple dimensions. Food, for example, varies along quality, amount, probability, delay, among others. Discovering how each dimension affects behavior, separately and in combination, is a difficult but critical task if we are to understand naturally occurring behavior. Suppose we have a choice between 2 behaviors, B1 and B2, for example, dining at Chinese or Italian restaurants, in a classroom, engaging in on-task or off-task behaviors, calling John or Mary on the phone for a chat. Behaviors B1 and B2 produce reinforcers, rewards at rates R1 and R2 respectively. How often we experience excellent meals at each restaurant, receive attention from classmates for each behavior, or have comforting conversations with each friend. What determines how we choose, or more specifically, how often we engage in one or the other behavior? the usual steady-state result accords with Baum's generalized matching law. The response ratio B1 over B2 is a power function of the ratio of obtained reinforcers R1 over R2, with S as a sensitivity parameter and B as a bias parameter. In some cases, B and S equal 1, which is Richard Ernstein's initial formulation of his famous matching law. Matching occurs in many contexts, including, surprise, surprise, chess openings. Just came out. The choice of chess openings is well described by the generalized matching law. But suppose that the reinforcers for behavior B1 and B2 vary along the dimension of amount and delay. In fact, that they trade off amount for delay. B1 yields more reinforcers than B2, but those reinforcers are also more delayed. All else equal, we know that the more is better than less, and immediate is better than delayed. But which is better? More but delayed, or less but immediate? Does a child prefer 2 marshmallows after 5-minute waiting period, or 1 marshmallow immediately? And how does value, and hence choice, change as the delays elapse? These questions are at the heart of research on self-control and impulsivity. Behavioral psychologists have studied empirically how choice depends on the magnitude of the options, the choice context, prior learning experiences, and the species and developmental stage of the organism. They have also developed theories and models to predict preference, the reversal of preference, and how to prevent such reversals in applied settings. One of the distinctive features of behavioral psychology is its historical approach to the explanation of behavioral patterns. Instead of moving in space from without to within the organism in search of inner cognitive or neurophysiological processes that generate behavior, the behavioral approach moves in time from the present to the past behavior-environment interactions in search of ways to understand how current behavior resulted from the integration of the effects of recurrent causes, each perhaps with a small effect operating over long periods of time. The behavioral explanation is not unlike the historical explanation of an adaptive biological structure such as the vertebrate eye in terms of the cumulative effects of natural selection acting consistently over long stretches of time. A correct behavioral account brings with it the immediate benefit of identifying how to implement specific kinds of behavior-environment interactions to promote desirable behaviors and reduce or prevent undesirable ones. A case in point, surprise, is the good behavior game, a remarkably simple behavioral strategy that implements group-based contingencies to reduce disruptive classroom behavior and promote academic engagement and pro-social behaviors. In its simplest version, teacher and students identify the rules that would make the setting a good place to learn, more enjoyable and pleasant, as well as the rule or fouls that would interfere with the desirable outcomes. The game is played at specified intervals like math class or reading class. The group is divided in 2 or more teams, and the team with the least number of fouls wins the game. Alternatively, any team with the number of fouls below a criterion wins the game, a group variant of a reinforcement schedule known as differential reinforcement of low rates of behavior. Highly visible scorecard shows daily the fouls and wins of each team. Developed in 1969 by Barish, Saunders, and Wolf, the good behavior game has been studied extensively since then. Its positive effects on student behavior were immediate. Other positive effects, however, were revealed only in long-term follow-up studies. In 2002, Embry remarked, quote, "The game worked pretty well and became a behavior modification trick most graduate students in behavioral analysis or special education learned during the heyday of behavioral psychology. Neither Barish, Saunders, or Wolf, nor the graduate students who learned to use the game as a classroom strategy had the slightest idea then how powerful the strategy might be for changing the future of children destined for multiple lifetime problems of substance abuse, violence, and school failure." In 2001, Kellum, a collaborator, summarized a large-scale randomized controlled trial study that tested the game with first and second grade children with a follow-up at ages 19 to 21. Quote, significantly lower rates of drug and alcohol use disorders, regular smoking, antisocial personality disorder, delinquency and incarceration for violent crimes, suicide ideation, and use of school-based services among students who had played the game. Several replications with shorter follow-up periods have provided the similar early results. Time to end. The influence of behaviorism remained strong until approximately the last quarter of the 20th century, and then it faded away like the voice is fading. Yes. Some historians have announced its demise multiple times. Most recognize that all scientific psychologists are behaviorists of a sort, for they all share behaviorism's commitment to objective methods and measurements to draw inferences about learning and memory, for example. But they also report the triumph of cognitive psychology and its dominant status for the last 40-odd years. The mind returned to its proper place, the center of psychological inquiries, and consciousness was restored to humans and animals, perhaps even to organelles. Psychology adopted a new root metaphor. The mind is a computer, and information processing became its lingua franca, at least in experimental areas. At the same time, few psychologists declare themselves behaviorists or behavior analysts, and most influential laboratories that once investigated animal learning and behavior have closed, their space promptly occupied by neuroscience research. There is a deep irony here. On the one hand, psychology remains a highly fragmented science, perhaps more fractionated with each passing decade. Currently, the American Psychological Association lists 54 divisions, a befitting name to its components. Anthropologist Clifford Geertz described the view. From the outside, at least, psychology does not look like a single field divided into schools and specialties in the usual way. It looks like an assortment of disparate and disconnected inquiries classed together because they all make reference in some way or other to something or other called mental functioning. Dozens of characters in search of a play. On the other hand, behavioral applications remain unrivaled, even after 4 decades of cognitive psychology. De Hoover and Hughes summarize, classical and operant conditioning form the core of treatment programs that are dramatically improving the lives of millions of children and adults living with developmental and intellectual disabilities. They are shaping people into better parents, teachers, colleagues, and friends. They are being used to radically cut drug and alcohol use in people struggling with addictions. Concepts and procedures derived from the psychology of learning are the beating heart of many clinical therapies used to treat phobias, panic and anxiety disorders, and chronic pain-related issues, as well as psychosis, depression, and substance abuse. The same ideas are deployed to train disobedient pets and improve the welfare of zoo animals and livestock, address health behaviors, help athletes improve their performances, and pathological gamblers beat their addictions, promote recycling and reduce pollution, and much, much more. The takeaway message is that the procedures, effects, and principles of classical and operant conditioning drive human behavior and can help us save lives and improve well-being. Ironic indeed. Perhaps it is time to see not the paradigm shift, but the paradigm lost. Let me close with the words of one of Skinner's last graduate students, a man of remarkable scientific creativity, Charlie Catania. We are all behaving organisms, though we may not like to be reminded. Like those unwilling to acknowledge our primate descent when confronted with the facts of evolution, some of us may resist the idea that our behavior is shaped by environmental contingencies. But if our discipline is to operate as a science, this cannot be cause to avert our eyes from it. And then, echoing the last paragraph of Darwin's The Origin of Species, he adds, there may even be grandeur in this view. It is my conviction that behavioral analysis will come to be viewed as a major scientific advance of the 20th century, comparable to that of natural selection in the 19th. The role that psychology will play in what follows from that advance remains to be seen. As for me, I would say that if psychology is ever to be a unified science of practical use, then it must be behavioristic. What else? Never mind. Thank you.
Well, thanks a lot, Armando. Really inspiring presentation for thinking and discussing. And let's start with the audience and see any comments. As always, every time you talk, I feel like shaking. And you may or may not agree with Armando, as I always said when we were colleagues at the University of Minho, but you are always going to be moved by his words and his statements. Thanks for your presentation here, and let's see if there's any questions. See Marco over there. Please, Marco, would you mind about name and affiliation, please?
Okay, thank you. Good afternoon, Marco Vasconcelos, Aveiro. I saw a lot of retrospect, not a lot of prospect. Looking at your title, in your title, I was expecting more prospect. And I'd like you to elaborate on that, particularly linking it to the earlier discussion this morning. The first introduction, our colleague said, well, look at the panorama in the psychology departments in Portugal. So let's link that to that discussion. How is the field in the country, if it exists at all?
Thanks, Marco, for the question. I didn't know what to leave out from the retrospective vision to include more the perspective. And also, because I don't want to depress you. I'm pessimistic, very pessimistic. In fact, that's what I had at the end of the talk that I had to remove for shortage of time. Time is the big constraint, right? Reduced expression of behavioral analysis, both basic or applied, that's a major problem. But I think Portugal has a much more serious problem. It's experimental psychology in general. So the reduced expression of all fields of experimental psychology in psychology department. I'm glad that George gave us some stats this morning. Undergraduate teaching by applied psychologists. That's a serious problem. When applied psychologists are teaching specialized areas, they know little about it. Training of master's students in behavior therapy, cognitive behavior therapy, and so on, is not anchored in the psychology of learning. Because there are no graduate programs, PhD student training is restricted to their lab research topic. Many psychology PhDs know almost nothing of their field. Sorry, it's the way I see things. We're forming many PhD students that make of ignorance a virtue. A complacent attitude of the accreditation agencies. They tolerate all of this, okay? 20 years or more since they exist. Policies of FCT, our National Science Foundation. I mean, we are all aware of the letter jump-started by George that we sent to FCT complaining about the shortage of funding, the bias in the panels, and the evaluation criteria and how they promote mere trendiness. What counts most is making a good immediate impression, a case study for self-control researchers, in fact. And, but I think there is a deeper issue. Far more important, I think it's the quality of our leadership. I can only summarize my pessimistic view by quoting John Burnham, a historian of medicine, that mentioned something that I think is befitting the current leadership that we have from Minister of Science after Mariano Gago, presidents of FCTs, and even university administrators. There are exceptions. Even, I like to praise Mariano Gago for his many initiatives to promote research in Portugal. But since then, I don't see anything. And certainly at the university level, but even in heads of department, okay? In a highly fragmented technical system as we live today, people flourished professionally who in another day would have been handicapped by insufficiency of breath. To say nothing of their lacking the culture and calling of Victorian scientists who argued for science because it was culture. So I'm pessimistic. I don't know. The numbers are short. So thank God I didn't prospect a lot.
Thanks, Armando. A follow-up by Michael.
Okay, that's a general view, I assume, about experimental psychology. What about the field of learning? Worldwide, in a sense.
Okay, so the problems will not go away because we don't look at them or we don't do good research on them. And so I believe that our contributions have been so far unrivaled. And show me if I'm wrong. And so the attempt to go into this, by the way, was the basis of a paper that I just co-wrote with a friend for an encyclopedia. First time when I was invited to write a paper on encyclopedia, I said, gosh, no, not encyclopedia paper. On the other hand, the person that asked me, that invited me, was my former supervisor, good friend, and a man I admire greatly. You saw the picture of John Staddon here. And so I decided to embark on what was the most difficult paper I've written in a long, long time. Because it forced me to have a sense of history which is lost. I would dare to say that most graduate students in psychology don't know what we're talking about in sufficient detail for a PhD. That's the first thing. It's not popular. Even in the States, the advent of neuroscience, with all the positive things that it has been done at the detriment of psychological research. So, unfortunately, it should not, but it is a zero-sum game, where the victory of some is the defeat of others. So, for example, the department where I worked, the animal learning people are gone. And it was invaded, if you allow me the metaphor, by a neuroscience research. It's fashionable. Psychology has always embraced fashions, trends. So be it. Okay? So I expect, to answer your question, this will be like a temporary eclipse. Okay? Because the problems will not go away. And we are doing things that nobody else does. And I have seen much good neuroscience research on learning, but I have seen also very bad, from the psychological viewpoint, science on learning. So we will be, I hope, a commodity, a rare commodity that, at least in the best labs, will find some niche. The problem, Portugal is a more serious problem because of its dimension, because its lack of tradition, not only in this area but across experimental psychology. So of the two, I'm much more concerned with experimental psychology. Okay, and so that's my 2 cents of wisdom about the field of learning. It's in a temporary eclipse.
Thanks, Armando. Let me also follow up. Sorry, go ahead.
So it's not really a question. It's actually, I guess, a reply to your last comment. In a way, for me, for instance, so I agree with what you're saying, and for me, it is strange for you to mention that, let's say, behavioral labs are replaced by neuroscience labs, because for me, behavior is a complete part of neuroscience. I mean, I don't see neuroscience outside of behavior or behavior outside of the neuroscience. And it is a shame that people like me, biochemists who never learned anything about behavior, are the people doing behavioral experiments now in the lab.
That's the
and
point.
Because we're probably not being able to take full advantage of the link between what we are doing and what you would know, or people much with a completely much deeper and broader view of the behavior would have. So I guess trying to so I think the idea is that it really needs to be multidisciplinary, and this is what I guess was the theme of this session, right? How we should interact much more. This has happened in the human genetics, for instance. This has happened also at some point. Somebody told me, oh, you know, clinical research is dead. There's no longer clinical research. And then we go to the genes, and we want to connect the genes to something, right? And we need clinical research. Again, that's why I was talking about the next generation phenotyping of humans, of animals. We are learning about the genetics in great detail, but then we need to fit this into solid knowledge on the behavior side. So for me, I mean, it's just narrow-mindedness not to see this like that. So just to support your...
Patricia, I agree completely, and I would say I'm all for multidisciplinarity, but equal respect. At this moment in time, I cannot get a job in the situation that you're describing, because they will always prefer a neuroscientist doing learning, knowing a little or a lot about learning, because there are some that do know a lot, okay? So if you want, I'm addressing a political issue political related to science. Why should an area be condemned to disappear because another area, at Duke, for example, we had extreme history of major contributions of animal learning, and then they hire people that were experts on the anatomy of retina. On the anatomy of a retina in the psychology department. Nothing wrong with studying the anatomy of retina, but why should it be at the cost of people studying? And don't tell me that both can coexist. Yes, if you have unlimited funds, but if you have choices and you have to make a decision, why do you go? It's not unusual. In biology at Duke, they always complain, the guys like Steve Vogel that did the whole organism biology, complain that everything is molecular nowadays, and so people like Schmidt-Nielsen could not get a job anymore, or Steve Vogel, the guys that did whole organism biology, had trouble. So other science also have these fads, but I fight for my Dulcinea.
João, just for the sake of transmission, you guys introduce yourselves, just name and affiliation.
João Peça, University of Coimbra. I just want to share what I think could be a little nugget of hope, right? One individual I respect quite a lot, and I'm sure you know him, Dale Purves. Started his career as biochemist, cellular biologist, very deeply involved in the neuromuscular junction, so really going for processes of at the cellular level, and then was involved in the creation of the neurobiology department, but he was actually then moved into a different field of illusions, which is, you know, more of a cognitive field, let's say. So could it not be made the case for greater mobility between the different areas? One theme that concerns me is that could it be that there's almost an exclusion of people within the psychology field that push themselves outside of the field of neuroscience? I would like you to comment on this. How do you see this interaction almost as a competition like you're suggesting, rather than, you know, a virtuous cycle?
The facts are on my side, João. I agree with your message. I think it's a hopeful message that in the near future, but in the present or recent past, I attended once the NIH panel in the United States, the behavioral thing, and most of it was neuroscientists. And they say what FCT sometimes says, well, it's, you know, the number of submissions. So they increase from coming from a particular sector of people, and a panel has to mirror those, has to have the same proportions. I agree with that speech that you gave, and I think a reasonable person with a sense of history would have the same thing. The facts are not that. You cannot get a job. Why are people closing their animal learning labs? Is it because they made no contribution? What I try to show is that they made no shortage of contributions, perhaps no less than any other area in psychology. And if I am wrong, please correct me. Please correct me if I'm wrong. Okay? We have had this author use after his book on cognitive and behavioral approaches, in the end says, we have had 40 years of cognitive psychology, and the applications are nil. Nil. Don't tell me about cognitive behavior modification, because that cognitive psychology is not the cognitive psychology that we are talking about in terms of information processing. Okay, it's not the same thing, although the label is the same. So I would go for that sharing, cooperation. I would love, Ana Pinheiro once, we sort of had a project of collaborating. There are many tensions. There are many deep problems of reductionism and how to interface, how to coordinate a neuropsychological neuroscience approach with a psychological approach. It ain't easy. It ain't easy. Okay? So the best minds have tried. So I'd love to collaborate on that, but this means collaboration, not at the cost of jobs of some and closing. England closed. In fact, as Óscar was reminding me, one of our evaluation panel, because I was there, from England, why do you guys keep an animal learning lab? We closed them all. Well, so much for you. That's the immediate reply that they should get, a sense of history. The same thing with the panel that evaluated the William James Center. In fact, when Marco and I spoke about what we did, I forgot, Marco, the order. We were talking about our research with pigeons, and they said, why do you study number, the sense of number in pigeons, or the sense of time for that matter? And I replied to her, she was from Hungary, Hungarian, right? The president of the panel. I gave the same argument that John Watson gave in 1913. If I'm interested in human language, should I study Hungarian only? Why shouldn't I study? I wouldn't know enough about language if I restricted to Portuguese or Hungarian. And so the same thing, why is it bad that I study animals? But the fact is that, look at the psychology departments here. See if they hire one. Okay? It's much more easily that they fire one or they invite him to leave than that they hire one. So it's a matter of fact. I talk with vice deans and head of departments, and I think generally, with notable exceptions, the next speaker is one of them, but notable exceptions, what I've seen at the administration is mediocre points of policies and mediocre people getting power. And when you combine mediocrity with power, you have an explosive combination.
Well, thanks a lot, Armando, and thank you all. This is the kind of discussion that we want to nurture. I really believe that's the big question, is how to combine identity with interdisciplinary outreach. That's not easy, as we have been discussing. And so let's move on right now to Rui Oliveira. Professor Rui Oliveira is currently a full professor in psychobiology at ISPA, Lisbon, and principal investigator at the Gulbenkian Science Institute and adjunct faculty at the Champalimaud Neuroscience Program. He graduated in biology from the University of Lisbon, where he completed a PhD a few years later. Rui Oliveira is past president of the Portuguese Ethological Society and was the founder and chief editor of the journal Acta Ethologica. He has been involved in research within the field of behavioral neuroendocrinology and behavioral genomics, particularly in neuroendocrinology and genomics of social behavior, the evolution and mechanisms underlying behavioral plasticity and comparative social cognition. Rui Oliveira led ISPA as a rector for an entire decade, and he was instrumental for creating a fertile niche for interdisciplinary research on animal learning and behavior. He will be presenting today on fish and flies, using model organisms to uncover the mechanisms of social cognition. Thanks, Rui, for being with us today, and welcome to our series. Hope this is just the starting point for future collaboration.
Okay, thank you for the nice words and the invitation to be here. It's a pleasure to be here and have the chance to present part of the work we do in the lab. It's also quite a responsibility after Armando's remarks on the field to step up and try to present work on animal behavior, basically, and neurobiological basis of this behavior. So what I brought here today to share with you is work that we do in the field of social behavior. So our lab is focused in trying to understand the mechanisms of underlying social behavior. Social behavior is ubiquitous in the animal kingdom. We are very social. A number of multicellular organisms are very social. Even unicellular organisms are social and also interact in interesting ways that can be modeled and studied. And in our lab, we are trying to understand both how organisms become social and what are basically the decision rules they need to implement to navigate the social world in which they live and why they become attracted to being with others. And also what happens in consequence of these interactions with others to their organisms, so how social interactions feedback on biological systems and have implications in terms of health and disease. In the lab, we are now using 4 different model systems. 2 of them are model organisms, and these are the ones that I selected for today's talk. Zebrafish and Drosophila. They are very handy because there are a number of genetic tools available that help us to dissect in detail the mechanisms underlying the behavior of these two animals. And then we use also other models that are more ecologically relevant and that we study in a mix of field and lab work, and these include the cleaner rest that we use as a model to study cooperative behavior in collaboration with Redouin Bouchard's lab in Neuchâtel, and the field work is done in nice places, so in Australia, in Lizard Island. And Africans. Going on time you see. But for today's talk, rest in Drosophila. And for those of you that are a bit skeptic that we can use these very dull animals to study social behavior, I'll try to convince you of that it is possible. So the first question is to see to what extent the mechanisms that underlie the use of social information are evolutionary conserved in biology, and this is a concept that came from Ivo Divo, from evolutionary developmental biology. There's this concept of genetic toolkits, that there are these genes that are evolutionary conserved across very distant taxa, and that they basically play the same function in these very different taxa. There are examples like Hox in terms of defining morphological territories in the developmental embryo, PEC6 for eyes, and so on. And in terms of behavior, despite the huge diversity in social behaviors, if you see it by their kinematics, by the way behavior is composed of different postures over time and space, but if you think about the function and the rules underlying the expression of these behaviors, there's a molecule that has been repeatedly implicated in the regulation of different aspects of social behavior, that is oxytocin. And so the first question is to try to see to what extent we can see oxytocin as a genetic toolkit for sociality, and if you can go back to a line of vertebrates that diverged from the line that we are part of, the tetrapods, And so if you can go back to a fish, in this case zebrafish, and if you can try to see if very ancestral types of social use of information are being modulated by oxytocin as they are in rodents and primates and humans. The second question has to do with social learning being domain-specific or general domain mechanism. This is a debate that has been ongoing in the field, and we try to address this using the tools that we have in Drosophila to uncover the genetic architecture and the phenotypic relations between the two of them. So the two stories that I will share with you today are trying to address these two questions. So starting with the first one, our idea of using zebrafish to study the social use of information is the fact that in zebrafish, and not only in zebrafish, but in a number of different fish species, when an individual is wounded, there's an alarm substance that is released from the skin, and it induces a very stereotypic alarm response or distress response in the conspecifics that smell that alarm substance in the water. This was first described by Karl von Frisch, one of the Nobel laureates from classic ethology. And so you can see, oops, let's see how I play the video. You can see in the video if it goes on. Yeah, you can see in this video what happened. So you see a zebrafish just in a neutral behavioral state in the tank, just swimming back and forth, and then we just add some drops of alarm substance into the tank and immediately responds with this erratic movement that is followed by freezing, and the video is still going, but the animal is motionless. So this is a behavior that is very easy to quantify if you use parameters like speed and depth in the water column. Over time, you can see that they come near the bottom, and there's a burst in speed that then decreases, and they freeze. So it's very easy to quantify these various stereotypic behaviors. And so our idea was that since they respond in a very stereotypic way, we could use these to study to what extent this distressed behavior could be transmitted to others that do not smell the alarm substance, but watch others that had smelled the alarm substance, and so these others had the first-hand information, and if they could respond. And so we basically came up with this very simple setup to study social contagion of fear. If fear distresses you as a term, as a construct, use alarm, distress, delta state, whatever pleases you. But there's something in the conspecifics that is transmitted to others that observe them. So there's an internal state in the ones that smell the alarm substance that distresses them. Sorry, that changed in response to alarm substance, so they are distressed, and their distress is transmitted to the observers that just get distressed by observing conspecifics being distressed. And so we have this paradigm to study social contagion of fear. And so what we did was to use a number of genetic lines that we developed with CRISPR in collaboration with Gil Lefkowitz for the ligand and the two receptors of oxytocin in zebrafish. And so this allows us to basically test to what extent oxytocin is involved in this behavioral contagion of fear in the zebrafish. And as you can see in terms of erratic movement, so here these first two columns are the wild types, so these are siblings, but they don't have the mutation, and these two on the right side of each panel are the mutants, and then on the left you have the control, meaning that they did not observe conspecifics getting distressed, and the right bar is the ones that observe the conspecifics getting distressed. So as you can see along this line that I'm going through with my cursor, the two receptor knockouts and the ligand knockout controls, they all respond to the conspecifics in distress by also expressing distress, so there's contagion. But when you look into the mutants, none of the mutants respond. So this is, and this is not here, so I was pointing you to the wrong panel. This is erratic movement, so where you see the effect is in freezing. The erratic movement is not as consistent, but in the freezing component of the response, there's a very consistent response. And so what you can take from here is that oxytocin is necessary for the contagion to occur. So mutants where oxytocin signaling is not working, they do not have the ability to copy the behavior of others. Then we took advantage of the fact that we have a ligand mutant, so you can basically provide this ligand mutant with a ligand by giving exogenous oxytocin, and you can ask the question, if you give exogenous oxytocin, can you rescue the phenotype? And they do. So what this is showing is that if you inject the mutants for the ligand with oxytocin, now they can copy the distress of the others. So with these experiments, we basically show that oxytocin is both necessary and sufficient for the contagion of fear in the zebrafish. Then we wanted to know where in the brain was something happening that we could associate to what we were observing. And so for these, we looked into a number of brain regions that are known to be involved in the processing of social information. And we used PS6 as a molecular marker of neuronal activity. And out of those many areas in which we looked, there were these two areas, VV and VC. They are putative homologues of the lateral septum and of the striatum in a mammalian brain that have this interesting pattern associated with the behavioral contagion. There is a decrease in activity in the wild types that express the behavior, and an increase in activity in the mutants that do not express the behavior, suggesting that there's an inhibitory cell population regulating this behavior that needs to be disinhibited, and that is overactivated and therefore inhibits the expression of the behavior in the mutants. Furthermore, we used the coactivation matrices to look into functional connectivity across the areas that we studied, and to make a long story short, what we find is basically an imbalance in excitation and inhibition across these networks, and also a lack of integration in the mutant when it is exposed to the distressed individuals. There's using community analysis, we can identify a community here that in the mutants is not integrated with the rest of the other communities that are identified, and in the controls there was. So apparently oxytocin is acting on specific brain regions, but also affecting the overall pattern of this network that is activated in association with the expression of these behaviors. We then look, well, you don't see much with this light here, but this is basically to show that there are projections from the oxytocin neurons to these areas that I was describing you. So not only we find statistical association, but then in real brains, you can see that the cell bodies of the oxytocin neurons that lay back in the preoptic area project rostrally to the VV that is here, and so you can see the cell bodies here and the projections coming here. We also marked the dopaminergic neurons because we know that they also project in that area, so to help us identifying our target area. Then finally what we wanted to see was if this was just behavioral contagion or if there was something more than that. And so, in other words, are they just basically imitating in a motor fashion what they see in terms of perception action, or are they doing something more than that? And so to try to decouple that, what we did was to use video playbacks. And now in one screen, we are showing a fish that is swimming around and then has 3 episodes of alarm behavior, so to say, during the 5 minutes of the playback. In the other screen, the fish is just relaxed, swimming back and forth. Then there's a time delay, and in a test phase, the 2 screens show the fish relaxed, and so we are using this test phase to basically query our observer fish if he can discriminate the 2 fish based on what he observed before. And if he manages to do this, it cannot be motor imitation because both are doing exactly the same thing. If he can discriminate, that's because he stored something in memory that is now recalling to tell one apart from the other. And what he has stored in memory is the state, the behavioral state of the 2 animals that was different because it's exactly the same video. It's the same fish on both screens. So the only thing that changes is the behavioral output that conveys the internal state of one of the fish. And so I'll skip through these slides. This is to show that there are no deficits in attention if we use body orientation as a proxy for attention. So mutants are as attentive as wild types to the distressed conspecifics. During the observation phase, we basically can replicate the same results that we had before with the live demonstrators, which is good because kind of validates the use of the video playback for this purpose. And then in the test phase, what we see is that the wild types have shorter latencies to approach the fish that they had observed before being distressed. For us, this was a surprise. We were expecting, or we were hoping, that they would discriminate, but we would make the opposite prediction. We would predict that they would avoid the ones that they saw in distress, because distress typically is a signal of danger. If they are distressed, that's because they saw something threatening. But in fact, they swim towards the one that they had seen before in distress, and they express a preference. So they not only approach more quickly that one, but they also spend more time near the one that they observed before in distress. So as a summary for this part, what I would argue is that we can have now a case of whatever you want to call contagion. It's not behavioral contagion. I think it's more than that. People doing similar things with mice and primates call it emotional contagion. I don't know if with fish this is allowed. Oxytocin seems to be both necessary and sufficient for this to occur. The fear contagion is associated with a decrease of activity in a specific population that seems to be inhibitory, and now we are confirming that through the identification of these neurons with GAD and GLUT lines that allow us to do that. The oxytocin role on this contagion seems not to be due to a mere perceptual effect of deficit of attention, but rather to an effect on the recognition of the state of the other individual. So together, these results suggest that there's a deep homology on the role of oxytocin in the regulation of emotional recognition that goes from fish to mammals, and this has implications if you want to use zebrafish as a model for this type of complex behavioral phenotypes. And this also shows that oxytocin that is involved in other types of empathic behaviors that are much more complex in species that are endowed in more complex cognitive abilities, such as helping behavior or consolation, which is not what we are showing here. But what I think we are showing here is a more ancestral origin of these types of behaviors that seem to be regulated by the same molecule that in mammals then regulates these other more complex empathic behaviors. The second part, or the second story of the talk, has to do with social learning and with this idea that social learning can be domain-specific phenomena. And for testing this idea, we decided to use Drosophila. And we decided to use Drosophila again to take advantage of the tools that are available for Drosophila. So there is the DGRP in Drosophila, which is the Drosophila Genetic Reference Panel. This is a collection of over 200 Drosophila lines that have been introgressed and sequenced. So we know the genomic sequence of each of these lines. And then there's a core panel of 40 when you want to phenotype complex phenotypes. Usually you don't use the 200. And because we were phenotyping learning, which is a phenotype that is time-consuming to characterize, we went to the 14. So typically what you do, whatever you are phenotyping, it can be body size, learning, whatever, wing length. You just characterize each of the lines, and basically you can then have the lines ordered like they are here, and then you can basically try to see, because you have the sequence for each of these lines. You can run a GWAS on these and try to come up with associations between specific polymorphisms and the phenotype of interest. With the flies, the interesting thing is that we can go one step further and we can do functional validation, because we have tools available that allow us to silence the genes that pop up in the GWAS. So we can, after this step of the GWAS, come up with a candidate list of genes, and then one by one silence them and see what happens if we have a phenotype when we do that, and it's like a confirmatory step. So that's what we did. And for this, we had a non-social and a social learning paradigm. So the non-social was conditioning of oviposition site. There was strawberry and banana. You just put quinine in one of the substrates, and they don't like quinine. So after a while, they start going to the one that has no quinine and avoiding the one with quinine. And so you can study their performance on these. And then for the social, basically, you have demonstrators that were conditioned. Then there's a demonstration phase where the observers observe the demonstrators avoiding one substrate and going for the other. And then in the test phase, you basically ask the observers where they would like to lay their eggs. And this is a very practical paradigm, because although I would say almost a behaviorist, or at least an ethologist, you don't need to see or record behavior. You just count eggs after the end, which is a consequence of the behavior they were expressing. And just by counting eggs in each substrate, you know what they did. And so based on that, you can compute a learning performance index, and then you can do what I was showing you before. It's usually done with this DGPR panel. And so what you can see here is the different 40 core lines ordered from the ones with the lower learning performance index to the ones with the highest one. If they have this type of values, it means that they are learning the opposite of what we were expecting them to learn. okay? So these ones, these lines here to the left, are the ones that apparently discriminate between the substrate with quinine and without quinine, but still prefer the quinine. So these are unexpected. And then these ones here are the ones that are not significantly different from the chance level, so they are the non-learners. And in red, you have an outbred population. In this case is the Canton S Drosophila population that we have used just as a reference population that we can see in comparison to it how the others are. We did the same thing for the learning. So you see that there are more lines that don't learn if the learning is social. So apparently social learning is, I don't know if more demanding is the term, but at least they are not as successful as many lines as they are for the social. But we still have the variation that we need to run the subsequent analysis. And the interesting thing, the first interesting result for me, it was this one. So when you plot their performance in the two learning tasks, there's no correlation. So the phenotypic correlation that you'd expect if that was a shared mechanism is that the ones that learn well individually would be the ones that would learn as well from observing others. That's not the case. So we don't find that correlation. The Canton Hess is there marked in red. What we reasoned then was that we could divide these in different quadrants depending on the performance indexes being high or low in social and social. So we can find lines that are good social learners and poor individual learners and the reverse, and the ones that are just non-learners, they cannot learn, and the ones that can learn whatever the protocol. And so from the phenotypic correlations, the message is basically that the two types of learning seem to be independent from each other. Then we went into the phenotype, to the genetic architecture to see if they would share the same genes. So we run the GWAS that I mentioned to you, and that Patricia already showed some results like these ones in the morning session. So you have here the Drosophila chromosomes and the different variants for each chromosome, and then the ones above this line are the ones that are statistically associated with our phenotype. And as you can see, for a social learning, there were 33 significant associations that matched 28 genes. There were 29 SNPs, 1 deletion, and 3 insertions. And these are the top 6 genes. I don't think that the genes by themselves are relevant at this stage, but this is just to tell you that we can find genes associated with the social learning. Then for social learning, we found a shorter list of variants that are associated, 13 that match 13 different genes, and these are the top 6 genes. The interesting thing is that there's no overlap in the gene list for the social learning and for the social learning. So the top genes, or the ones that are significantly associated with our phenotypes, they do not overlap. So again, supporting the idea that we have different processes ongoing. Then the last step that we are now doing is to do the functional validation, and the functional validation looks like this. So this is one of the genes for social learning, the glutamate receptor. And so you can see that here on the left, silencing the gene improves social learning, but it doesn't affect the social learning. So the social learning is not affected, and a social learning with which it was statistically associated is affected. And an example of a gene that was associated with social learning, where the reverse basically occurs, that there was no difference in the social learning, but it is confirmed its involvement in social learning. So with this, we are basically showing that we can use a species that is apparently as simple as Drosophila to study social learning. And using it, we showed that there's no correlation between social and the social learning in Drosophila, at least in this paradigm. Then the GWAS indicates different genetic architectures for the two types of learning. And the functional validation so far is supporting these top genes as being functionally involved in each type of learning, but not in the other. So together, these results suggest that social learning seems to be domain-specific in Drosophila, although I think this should be taken carefully. I think this discussion of things being domain-specific or of general domain, it depends on the depth of analysis that you will use. With this, I hope to have basically showed you that we can use model organisms for which we have genetic tools available to address mechanisms underlying complex phenotypic traits in behavior. That oxytocin seems to play an evolutionary conserved role in the ability of individuals to use social information from others, and I think this is a very basic feature of sociality in vertebrates, and all the more empathic behaviors that we are so proud of as humans. I think they have their origin here, and that social learning seems to be domain-specific, lacking a phenotypic correlation with the social learning and displaying a different genetic architecture. This is the finishing slides to acknowledge all the people in the lab involved in this and all the collaborators. In red, you have the names of people in the lab, current and past lab members that did the work on the zebrafish in collaboration with the lab of Gil Lefkowitz at the Weizmann Institute in Israel. And in green, the Drosophila people, also in the lab and also in collaboration with another group at the IGC and with Luisa Vasconcelos at the Champalimau. And I would end with an advertisement that we are looking for a new lab member. So we have an opening position for a PhD student with salary. And so if you know of someone interested in, going, in fact, is to work with cichlids, so there's the opportunity of going to Tanganyika Lake and dive in the lake. Also do a lot of bioinformatics in the lab. But if you know of someone or if you are interested yourself, just drop me an email until the end of the month, and thank you for your attention.
Thanks a lot, Rui. Any questions, João? So that's João Peça from the University of Coimbra.
Thank you for the amazing talk. First of all, I think following Armando's bleak prospect, I think it's fantastic to see such amazing behavioral studies being performed. So regarding the first part of the work, I have one question for each side. For the first part of the work, you use the shoals, basically a larger social group.
Something there. Well, I can't remember the name. But basically it's like voting, when people are voting and you are influenced by how many people are voting in one direction, that you can follow that number of people. But so we were addressing two things and none of them worked. So one, we call it salience, which is the number of fish that are doing something. And the other thing that I cannot remember the name is you have the same number of fish making a decision, but they are deciding different things. And so you have to basically, well, this is annoying. I can't remember the word. And you have to go with majority, okay? And it's one of these odd situations where the data was very weird, and we decided that we need to repeat it. So that's something that we have tried. The first results were strange. So honestly, it looks like somebody mixed up something, but since nobody assumes that something was mixed, we basically don't trust the data, and we will need to repeat conformity. It's the word that I was missing.
So you're saying that one of the parameters that may vary is the congruence, right? If they all are going in the same direction, or some of them are...
So the salience is having 1, 2, 4, 8 doing all the same thing, but it's the number of fish that changes, the number of demonstrators. The other thing is that you have the same number of demonstrators, but a portion of them are doing the distress behavior. And that's the conformity part. The other one is what we call the salience part.
Very much looking forward for...
So the salience, we are sure that the number of fish doesn't seem to affect.
Okay.
The conformity, it seems to have a signal, but it goes in the opposite direction from what it would be expected. So we really need to repeat the experiment to be sure.
And so regarding the second project that you showed us, at least one of the genes is very familiar, the NMDA receptor. So it's one of these proteins that is supposed to have a broader function regardless of the brain region where it's being expressed. So mechanistically, how do you see the differences between the social learning and the non-social learning at a mechanistic level? Do you think it's perhaps specific brain areas that are being affected, but then how do you reconcile that with something as, you know, as broadly important as NMDA receptors being affected, playing with that?
So I'm not giving another talk, I'm just going back to share this with you. So what we are taking advantage of by using Drosophila at the RNAi approach with the GAL4 UAS lines is that we can use promoters. So right now we are using pan-neuronal promoters. So it's just we are, you know, shutting down the gene in whole Drosophila brain. For the ones that we have hits, for the ones that we confirm their functional involvement, then what we have already in the lab is specific promoters for specific brain parts or cell populations that will allow us now to dissect where these genes are acting. And I have not mentioned that because I thought it was a detail, but you picked the detail, so that's where we're going.
Would you mind about introducing yourself? Name and affiliation, please.
Okay. So I'm Art Pilaczynski. I'm from Jorge Almeida's lab. Anyway, well, this is, yeah.
What should I do?
Slightly disturbing. Anyway, so I wanted to ask you if you know any, you mentioned collaborative behavior in the beginning of your talk, that you studied it. Do you know any non-mammal species that would be capable of collaborative behavior? Out of curiosity. Huh?
The cooperative behavior that I mentioned?
Yeah.
Yeah. I mean, the rest, the fish does it.
Exactly. How, so how does it do it?
Okay. That could be another talk. I'll try to be brief.
Yeah, do it
okay.
in 20 seconds.
In 20 seconds. So clean arrest, they clean clients. So there are client fish in the reef that come to the cleaning stations and they clean them. It looks like mutualism, but in fact there's a conflict of interest because what the rest likes is the mucus and the skin of the client, not the ectoparasites. But in order to have access to the meat of the skin of the client, he needs to provide the service against his preference, so to say, right? So he has to remove parasites, and then he gets a chunk of mucus and a bit of skin from the client. and then he needs to appease the client because the client now is mad, there was bad service, and so they do this massage with their fins that we show that reduces cortisol levels. This is a whole thing. We needed to travel to Australia with massage machine.
Does that involve collaboration between conspecifics?
Heterospecifics.
Exactly. So can you give me another example? I'm sorry for it is slightly off topic, but I was really intrigued by that.
Well, you have a number of cases. You mean cooperative behavior? When you say collaboration is cooperative behavior?
Exactly, and I mean like within species.
Within species, there's work done on rats, not by ourselves, but
Yeah,
you have pooling behavior in rats. You have similar kinds of things in primates.
Do you know a non-mammal species that would do that?
Within the same species,
Yeah.
but hence it's kinship. I think you refer to non-kin, right? Because the explanation then it's biologically.
But just the behavior first. Are there evidence of cooperative behavior within species, non-mammalian species?
Yeah.
So that's what he's asking, I guess.
Yeah, but I think the question has to do with non-relatives, because otherwise the explanation will be easy in terms of inclusive fitness. This explanation, evolutionary speaking, it's more demanding in terms of what's going on in the system. I'm not aware. Well, I'm aware of this one, but it's between species. So there's clients and service providers that are not the same species, and then they have a whole. It's like basically a game theory thing that they are doing, that you can manipulate the metrics, make them do things against their preference, blah blah blah. I don't know if this addresses the question.
Okay, well, it kind of does, because I was just wondering if there's some example that would be known to, people working with zebrafish, for example, or non-mammalian species that I would be missing. But okay, all right, thank you.
Well,
Yeah.
it just came to mind. There's one thing that can be seen as cooperation, which is, because now you mentioned zebrafish, so I'm not sure if zebrafish does it, but minnows do it, which is predator inspection. So when you have more than one animal doing predator inspection, they take turns, which is a kind of cooperation because they are, taking a risk of being caught.
Mm-hmm.
And so and now that I mentioned this, there's a lot of work on sentinel behavior in different species, a number of them mammalians. So, but at least minnows and sticklebacks, they inspect behaviors. If you put food and the predator between them and the food, they have to go there and inspect, and they take turns. And you can also model that in terms of reciprocal altruism, basically.
There's a question from, thanks a lot, there's a question from Armando.
I was curious about this alarm reaction that you described. So it seems to be biphasic. So the fish first increases the speed,
Yeah.
and then goes to the bottom,
Yeah.
and then rests.
Yeah.
I think about adaptive value of that. I mean, in a natural environment, you wouldn't see the second component, would you?
You do.
Just
You do.
escape.
So freezing, you see in the
So in the natural environment without boundaries.
yeah.
So what do you see?
The two components. Well, I haven't seen this in the natural environment myself, so I'm not claiming that. But in freezing occurs in mice, in many different animals, and freezing is seen as an adaptive behavior. So it has to do with basically evaluation that the animal does in terms of having an opportunity to escape.
But missing that opportunity, failing that opportunity, it freezes. A rat will not freeze right away to try to if it can run away.
In fact, we have work that it's not solid, so I tend not to show it, but there are individual differences. So there are some individuals that have a higher probability of freezing and other individuals that have a higher probability of doing active escape responses. So there's like an active and a passive phase, and we are trying to understand what drives one or the other. One thing that we noticed is that there's some inter-individual variation in the way they use each of them. So there are some individuals that are more prone to freezing than others. And we are now trying to link that to, that's another thing that you do in the lab, that is judgment bias and optimists and pessimists and blah, blah. And it seems to be related to the way they assess. So some of them are more negative about the outcomes of the outer world, and these guys tend to do more one thing.
So it's just like people, right? So my question was in the first part, you said you were surprised because they prefer in the choice phase to approach the alarm guy. Isn't that empathy then?
I was about to say that. I won't say that word. I won't say the E word.
Well, just a question.
Marco in the room.
Well, just a question. When you refer to your contagion paradigm, at the beginning you refer as a social contagion paradigm, but then on your conclusions you switch to an emotional contagion paradigm. So it looks like you are using a social emotional contagion paradigm. Have you attempt to dissociate both on the role of oxytocin on either social or emotional contagion?
Okay. So thank you for the question, because it allows me to make clear something that I think it's really important. So this is clear social contagion. Then the mechanism can be through a change in the state of the observer that makes it express the behavior that matches the behavior of the other, or it could be just motor imitation without, this matching in internal state. The evidence we have now is that there's a matching in internal state, at least as seen from, for example, levels of cortisol in the observers that match levels of cortisol in the demonstrators. And this is our first indication that I would dare to say there's emotional contagion and not only, behavior transmission of whatever. And also our last result that doesn't prove it's emotional contagion, but rules out that it's just motor imitation.
Thanks a lot.
Thank you.
Thank you.
Okay, so first of all, Jessica, can you hear me? Oh, yeah. Yeah, you can hear me, right? But we cannot hear you, though. Yeah, we can't hear you. Oh, there we go. We got it. I got it. Okay. First of all, sorry for the delay. It is my great pleasure to introduce Jessica Cantlon. Jessica is an amazing researcher, as you all know. In fact, she embodies some of the stuff that Armando was saying. She actually works with animals, or non-human primates, with humans, with adults, with children. She does neuroimaging, she does behavioral research. She is she does it all, and she's a fantastic researcher. She's also a great friend. She's the Ronald J. and Mary Ann Zdrowski Professor of Developmental Neuroscience at Carnegie Mellon University. She's known, obviously, for many things, including numerical cognition in human and non-human primates. Her work has been influential, immensely influential, in cognitive science. But that's not all. Jessica is also, or was also, the Time Person of the Year 2017 as one of the silence breakers with the movement Me Too, and she's an advocate for gender discrimination and gender issues. And so her work goes way beyond the stuff that she's going to present here today. Again, it's my great pleasure. She is a great friend and she's a fantastic researcher. It's great that you're here. Thank you very much. And the floor is yours, Jessica.
Okay, thanks so much, George, for the introduction. And I want to say thanks to you and Clara for connecting us with this incredible scientific community at Coimbra. Hold on one second. There's a slight delay in the audio, and so I'm just going to turn you guys down. Okay. Yes, and so I've really enjoyed the talks that I've been hearing so far, and I've engaged some of the questions that were raised in both talks, especially in the first talk, maybe in the discussion after my talk. And so I'm going to talk about math monkeys and the developing brain. My lab's interested in the origins of thought very broadly. The question that we've spent a lot of time on in my lab is this question of, where did human concepts of mathematics come from? So how did humans get these initial concepts of number and counting that have grown into a complex system of mathematics in us humans? And to answer that question, we take an interdisciplinary approach that bridges human and animal research. And so I'm going to talk about our comparative work of basic representations of number in non-human primates in young children and in people across cultures. And our work is motivated in part by the work of Nico Tinbergen, who won a Nobel Prize in 1963 for his work explaining the different causes of behaviors. And his argument was that you really need to answer all of these 4 questions here in parallel in order to arrive at an accurate and complete explanation for any behavior. And so in order to really understand both its causes and its functions. And so people often talk about Tinbergen's 4 questions as being the major defining categories for explaining behavior. So how did it evolve? And in my lab, we study this at the species level. How does it develop? So the effects of age, of experience, of maturation, of culture. How does it work? So these are the mechanisms, both the cognitive and the neural mechanisms, and what's it for? So what's the ecological utility of the behavior? And these 4 questions are important to consider together because your answers to all the others. So the answers are interdependent. What's it for affects how it develops, how it evolved affects how it works affects how it might develop or evolve from here. So these are all mutualistic constraints that determine what's possible in the human brain and in behavior. And because of this interdependency among these questions, Tinbergen advocated for the fusing of many sciences all concerned with one aspect of behavior into a coherent science. And so that's what we're trying to do, at least as much as we are able in the cognitive research program in my lab. And so we work across species, development, across different cultures, because these groups reveal different constraints on behavior. And so to try to figure out what causes what, it's useful to look at these multiple levels of analysis in parallel with the same questions, the same stimuli, same paradigm, same task. And one of the foundational findings that's come from this literature, not just from my lab, but from many labs, is that all of these groups shown here on this slide can estimate the numerical values of things, even without counting. So the numerosity of things. So this is a video here of a monkey doing a numerical task, and her task here is to first press that black square in the bottom corner of the screen to indicate that she's ready to start, and then 2 arrays pop up and she's supposed to hit the smaller number first and then the larger number. When she's right, she gets a juice treat, Kool-Aid, that's in the straw that she's holding in her mouth at the front of her chair. And we and others have shown that primates and other non-human animals can do really well on these kinds of numerosity tasks. They can perform with pretty high accuracy. And so babies and kids who don't yet know how to count can do this type of discrimination. Lots of people study this phenomena in children and related phenomena. Groups of people who don't have formal education and don't use counting in their everyday language are able to do this kind of discrimination. So here in this photo, here is a picture of a Tsimane woman from a group of people that my colleague Steve Piantadosi and I work with in the Amazon. So these people don't use counting or number words very much in their daily lives, and yet they're still able to do basic numerical computations, even in the absence of symbolic counting. There are similarities in how these groups perform these numerical tasks. So this is an example comparing numerosity discrimination for monkeys and adult humans when humans aren't allowed to count. So what's shown here is that accuracy during a numerical discrimination task, like the one in the video I just showed, is similar for monkeys and humans. So you can see that both of these groups generally perform well above chance. But you can also see that performance gets worse for both groups as the numerical ratio between the sets that they're comparing increases. So it's a lot easier for people and monkeys to do the task at small numerical ratios compared to large ones. So they're better at comparing values when the comparison is something like 2 versus 30 dots, which is a 0.1 ratio, than if they have something like 28 versus 30 dots, which is around a 0.9-ish ratio. So accuracy in this kind of rapid numerical discrimination depends on the ratios of the numerical values. And this pattern indicates that subjects are approximating. So they're using more of an analog type of numerical thinking as opposed to a precise or digital type of numerical thinking. This is a visualization of what analog numerical thinking is like. So what we hypothesized the internal representation of number is like for animals and people when they're approximating numerosities. So as the actual objective number that's shown to the animal, as that number gets larger, the probability of noise in the animal's or person's representation increases. So those distributions are getting fatter. And so then as a consequence, the overlap between representations, the probability of confusion between numbers also increases. And then the probability that an individual will have a subjective representation of that objective number decreases as those numbers get larger. And so that's why we see this ratio effect in performance on numerical tasks. It falls out of these proportional relations among these mental analogs. And so this is an analog number representation. This analog format is probably the most basic and primitive way that we have for reasoning about quantities. And the question we're interested in the lab is how could this kind of numerical representation help to establish human mathematics? And so to get at this question, we do these large scale comparative studies in order to try to understand the contributions of evolution, of development, and of culture to aspects of numerical cognition. And so in this first study we looked at, that I'll talk about, we looked at how purely numerical thinking compares across species, across age, and across cultural groups. So we trained monkeys and kids and adult humans to categorize stimuli into one of two categories, and the categories were a little bit versus a lot. And so during training, the subjects they'll see a stimulus and they have to press a button for which category it belongs in. So if they see a stimulus with a small number and cumulative surface area, they get rewarded with a treat if they press the category A button, which would be like the button on the left side of their screen. But if they see a stimulus with a large number and large cumulative surface area, they get rewarded for pressing the category B button, which would be the button on the right side of their screen. And they gradually learn these categories over time. And then during testing, we give the subjects novel values, so novel stimuli with new combinations of number and area values, and we see how they categorize them. So they'd be trained on these two categories, A and B, where category A has a small number and small cumulative surface area. Category B has a large number and large cumulative surface area. And then we test them with these novel values. So like a small number with a large surface area value, and like this, a large number with a small cumulative surface area value. And on these novel test trials, there's no wrong answer. They're told that they're right no matter what choices they make on these stimuli. They always get positive feedback. But importantly, if the subjects are basing those categories A and B that they learned during training, if they're basing them on surface area, when it comes to these novel stimuli, they're going to divide the stimulus space horizontally and categorize everything that has a small surface area as A and everything that has a large surface area as B. But if they're categorizing these things based on number, they're going to divide the stimulus space vertically this way and categorize everything with a small surface area as A and everything with a large surface area as B. I'm sorry, everything with a small number as A and everything with a large number as B. And so this vertical boundary indicates that subjects are using numerical representations to get through the task. And that's the kind of pattern that we see across these groups. So monkeys, 4-year-old human children, adults in the US, and shamane adults all have these vertical crossing category boundaries, showing that they're basing their judgments more on the numerical values of the stimuli as opposed to the surface area of the stimuli. And it's important that these shamane adults show similar behavior to the US adults because shamane people use counting and number words far less than people do in the United States. And so the similarity between the shamane and United States adults show that this kind of number biased categorization is part of human perception rather than cultural. We can also look at their category biases this way. So this graph here quantifies how much of a number versus area bias there was in each of the different groups that we tested. So this is showing the relative contributions of the dimensions of number and area to explaining subjects' category judgments on those novel stimuli. And what you can see is that all of these groups are showing a stronger influence of number over area in these judgments. The number weight in the behavior was between 2 and 4 times stronger than the surface area weight. The weight varies in strength across these groups. So adults from the US and shamane adults show the strongest number biases, whereas 4-year-olds and monkeys have the weakest. But despite the differences in gradation, we see that there's this developmental and evolutionary continuity in the primary dimension that animals and people are biased to use. And so numerical information is quite salient across these groups. So adults from the US and shamane adults show the strongest number biases, whereas 4-year-olds and monkeys have the weakest. But despite the differences in gradation, we see that sorry, I can't. I feel like someone's talking right now, but I can't totally hear.
Sorry, do you have your...
So the difference in the degree of numerical perception among people seem to depend a bit on culture. So we did see some variation across groups. And what we find here is that there's a significant effect of formal mathematics exposure, which is shown here on the x-axis, on how subjects quantify these stimuli, both in the US children and also in the shamane. And so even though, as you can see on the left, that the degree of number perception that subjects showed was correlated with both their age and education in these US kids, in the shamane on the right, the effect wasn't driven by age because they're all adults here. So here the effect was driven by the amount of education exposure that adult shamane people had in their past. So having
Jessica, sorry, can I interrupt you? Do you happen to have your YouTube on with sound?
I do. Yes.
Okay, could you turn that off?
Yes.
Sorry.
Hold on 1 second. But I think I won't. I'm not sure if I'll be able to hear you.
You will. No worries.
1 second.
Jessica, sorry, can I interrupt you?
Uh-huh.
Do you happen to have your YouTube on I think that's it.
Did it work?
Yeah, it works.
Okay,
Great.
sorry. This
No
is what I was trying to ask you when I was texting you.
Oh, sorry.
Okay, no problem. Okay.
Let's go back.
Okay, here we go. Back to the slides and back to, okay. And so here we're seeing these effects of culture on numerical processing. And so we see that some aspects of our ability to represent numerical values are affected by culture, and they appear to be acquired from this unique cultural experience that humans have. Okay, and so there are cultural effects on the degree of number, precision and salience in the representation. But very early in development, before some of those cultural factors can take hold, the representation of numbers seems to be driven by very general primate factors. And so we've tested infant monkeys on quantity tasks. So these were 1-year-old baby monkeys who were born at the facility and had never participated in an experiment before, and were in fact still nursing and just starting to eat solid food. The task that we did here was just really simple. We just baited 2 cups with some number of food pieces. The monkey just has to pick the cup that they want. Baby monkeys, infant monkeys, develop faster than human infants, and so they can sit and make choices at an earlier point in development than human infants can. The maturation of perceptual and motor development in monkeys is about 3 times faster than it is in humans. And so this graph is showing these infant monkeys' performance on the very first exposure that they had on this basic quantity task with a bunch of different number pairs at different ratios. And you can see that the infant monkeys are good at this task, so they're above 50%. And we found that they're about as good as 3-year-old human children on this task. So these data on the right here are from children who did a similar numerical task. And what this suggests is that the 3-to-1 ratio that we're seeing in maturation for perceptual and motor development in monkeys seems to apply to numerical discrimination too. And so what that suggests is that the bottleneck on the early development of numerical cognition in humans and in other primates seems to be a very general one. And it follows this relatively generic 3-to-1 ratio of perceptual and motor maturation. So this very early development of numerical sensitivity seems more maturational across primates and humans rather than experience-based. We've been looking at natural behavior in the wild to try to understand how different quantitative dimensions are related in nature. So there was this recent study in Science that used GPS collars on wild baboons to measure troop movements. And they showed that baboons make decisions about which direction they're going to move in democratically, using tallies of how many animals are moving in each possible direction. And we've been working with those data from the wild. And one of the things that we wanted to know was whether the animals are basing their decisions on the numerical values of individuals moving in these different subgroups, or are they just basing it on the total mass or sort of amount of stuff that's in each subgroup. So when a baboon comes to a decision point like this with 2 subgroups of animals heading in different directions, do they use number or mass to choose the larger subgroup to join? This, interestingly, is the correlation between number and mass in this environment. And so you can see that these are variables that are very highly correlated in nature. So there's about a 0.96 correlation between them. And this is interesting because it shows that there's a lot of data for the baboons to access in their environment about the tight relation between these variables. So in most cases, these 2 dimensions are going to give you the same answer about which set has more. So this correlation would be pretty easy to learn from the environment. But we found that despite that high correlation between these variables, baboons can treat these dimensions as independent. So in cases where there's a tie between number and mass about which direction to go in, and in cases where number is greater for one subgroup and total mass is greater for the other, the animals choose based on number most of the time, not on mass. So over 70% of their decisions go with the numerically greater subgroup. So we think that there's some evolutionary priority for numerical reasoning in primates, and that this ability to single out number as an actionable property of things is probably quite old. Most mammals and birds have some ability, and even invertebrates have some ability to represent quantity of some sort. And so we've been doing some of the first analyses across many species to try to figure out whether those abilities are evolutionarily homologous across species. That's unlikely, but meaning they have a shared evolutionary origin of some sort. And we're interested in the degree to which those abilities are under unique selection pressures across narrower clades. And so we've gathered data from many different researchers in the field who tested different species with basic numerical tasks. And we have data now from over 500 individual animals representing over 30 species of birds and mammals. And this figure here is just plotting the Weber fractions of the different species that we have represented. So here a larger value is worse because it means you need a bigger difference in numbers to tell them apart reliably. So a larger Weber fraction, it means you're worse, you're less sensitive. And there is some correlation between species Weber fractions, their sensitivity to number, and their evolutionary distances from one another. So closely related species have more similar Weber fractions than species who are farther apart on the tree. But there's lots of variability that's not just explained by phylogeny. And what that means is that numerical sensitivity is likely under different selection pressures across clades. And so evolutionary distance alone doesn't explain the data that we're seeing. And so we've started to look at aspects of brain morphology and ecology to try to get a foothold on what factors might relate to variation in numerical cognition at the species level. And so we've been looking at things like brain size and number of neurons and neural density. And what we're seeing is that across species, Weber fractions are related to brain size and particularly to species cortical and cerebellar densities. So a negative relation here means as neural density gets higher, Weber fractions are getting better because a lower Weber fraction is better. You're more precise. And so we're seeing that relation between neuron density and Weber fraction. We also see that domain general cognitive skills like reversal learning, memory capacity, those general cognitive skills also are related to a species Weber fraction. And these analyses are interesting because they give us clues. They point to both hardware and software constraints at the species level that help explain what's possible at the individual level. So this is evidence that the degree of precision in an individual's numerical cognition, if you just pluck an individual from any species out from the world, part of what's going to determine how well they can discriminate quantity is driven by evolutionary factors that are very generically related to their species. How are numerical representations implemented in the brain? So Alyssa Kersey from my lab, a PhD grad from my lab, has done a lot of this work that I'll talk about now. And she studied how basic numerical representations in human children compare to those that are observed in non-human primates and adult humans. And so we've done a lot of fMRI studies with human children at various ages, very early in development, and the kinds of paradigms that we use with these young kids during fMRI scanning are paradigms like these, where subjects passively watch stimuli, like in this video, and we measure their bold responses to changes in those stimuli. And here we're interested in brain regions that respond to changes in the numerical values of the stimuli, but not changes in other dimensions like the size or area or brightness of the stimuli. And the general finding here is that humans and non-human primates show numerical sensitivity in homologous regions of posterior parietal cortex. So the top image here is monkey neurophysiology results from Andreas Nieder and his lab. The bottom figure here is adult fMRI data from Manuela Piazza and Stan DeHaan and their colleagues. And these are our data from 3 to 5-year-old children. So these images are showing regions that were sensitive to numerical values by showing adaptation to repeated numerical values and also increased neural responses to novel numerical values. And so we see that numerical sensitivity in similar regions of the brain across species and between children and adults. The curves here on the right show the neural sensitivity of that brain region to numerical values in monkeys at the top, the 3 to 5-year-old kids from our sample, and adults from Manuela and Stan's data. And you can see that in monkeys, the tuning curve of number is similarly coarse compared to the one for 3 to 5-year-old human kids. So they both show this pretty crude tuning to numerical values. And in adults, you see that the curve of numerical sensitivity is much sharper and more precise. So all of these groups are showing tuning to numerical values in parietal cortex. That tuning gets sharper with age and experience in humans. When you look within this brain region in children, you also see a numerical bias analogous to what I was showing in behavior earlier on in the talk. So in these parietal regions that show this numerical sensitivity, this numerosity tuning, you don't see comparable tuning to any old dimension. So you don't see comparable tuning to cumulative surface area or color brightness. These other lines are just flat, showing that the parietal region that represents numerical values in these kids is not equally sensitive to other dimensions. And this is from a non-circular leave-one-out analysis in kids. And what this shows is that this region of parietal cortex is biased towards numerical processing in young children. It's not just modulated by any dimension. These neural responses that we see in this region in children are related to their behavior. So we found that when children passively view numerical stimuli during MRI scanning, their neural tuning curves parallel their behavioral performance curves. So these kids did a task outside of the scanner where they judged the relative value of 2 arrays of dots, which one was larger, similar to that monkey video I showed earlier. And so this figure here is showing those children's behavior at choosing that larger number across different numerical ratios. And so here a 1.0 ratio means that the numerical values are the same. And as you go out from there, the numerical values are getting more and more different from each other, the farther you get from one. And in both the kids' behavior and in their neural activity, we see these responses are systematically modulated by numerical ratio. And we found that these values were correlated at the individual level and that children showed a comparable, though not identical, sensitivity to number in both their brain and behavior. And so we think there's a good deal of both evolutionary and developmental continuity in this region of parietal cortex, the intraparietal sulcus, in human mathematics development. And we found that this IPS region is consistently recruited by young kids across a large range of ages, across many different types of numerical tasks with stimuli in a variety of numerical notations. And we see these kinds of number-related IPS activations in children relative to a lot of different types of control tasks, including tasks that are equally difficult and demanding. And we found that you can even see a relation between neural responses in the IPS and children's mathematical abilities when children are doing a task that they might do naturally in the real world, like watching Sesame Street. And so this is the last study that I'll just describe briefly. We do a lot of studies to try to understand the ecological validity of some of these functions in humans, some of these neural processes. So this is an example of a study where children and adults watched an educational video for around 20 minutes. So this was a Sesame Street video with 4 to 8-year-old children. This is an example of the neural data that we get from natural viewing. So these are the average adult and child time courses of neural activity. This is just from one small brain region from across that whole video viewing session. So this is neural activity levels over time. In red is children and in blue is adults. And you can see that the average pattern of neural activity here is vaguely similar between children and adults. The correlation here between children and adults time courses is about 0.52, so that's pretty strong. And again, these time courses are from just one region. We get one of these from each of thousands of different points in the brain, and each subject has his or her own time course for each of those points in the brain. And so we calculated this kind of correlation here between each child's and each adult's time courses at every point that we sampled in the brain. And then we tested where in the brain is the strength of a child's neural correlation to adults predictive of their mathematics ability. So we asked where in the brain individual children's degree of neural correlation to adults was predictive of their performance on a math test that they took outside of the scanner. And this is what we found. So this image is showing in green the points in the brain where the strength of children's neural correlation to adults is predictive of their mathematics performance. And so you can see that this analysis, which could have turned up any region in the brain because it's a whole brain analysis, showed a significant relation in this same region of parietal cortex that we see for these more basic numerosity tasks across species. So what this shows is that the degree of similarity between a child's neural response in the IPS and an adult's IPS response while they're watching this educational video predicts how well those children will perform on a math test. And so this means that the maturity of children's neural responses while they're doing something that they might normally do, like watching Sesame Street, is related to how they understand mathematics in school. And just to bring this full circle, we also tested these same children from this Sesame Street math study on a basic numerosity task in the scanner with arrays of dots. And we found that the neural regions that children recruited during that basic numerosity task overlapped the neural regions for mathematical processing that emerged from this analysis of the Sesame Street math video. So these converging results, which come from two very different tasks, one that's very simple and controlled, another that's more naturalistic and unconstrained, the convergence between those results gives us more confidence that what we're looking at is the developmental basis of numerical processing in the human brain. And so this series of studies is an example of how a mechanism that's quite old can become hijacked by human development and cultural influence to do something extraordinary, like learn formal mathematics. And I just want to thank you all for coming today, and I also want to thank the people in our lab at Carnegie Mellon who conducted many of these studies. Thank you.
Okay, thank you, Jessica. Are there questions from the audience? So there's one question there. André Pérez. André.
Hi. So I'm André Pérez from Universidade de Coimbra. So thank you for the fantastic talk, Jessica. So I was wondering to understand a little bit more about that figure, that graphic that you showed, about the correlation between the difference when the people must to choose between two different groups, the numeric groups. So you have a U-shaped graph, so as different as the groups. So for instance, we have a group with 2 points, another one with 10. So it's easy to separate them. So we have a bigger or a smaller activation, I guess. So a bigger activation. Yeah. So a bigger activation. So what activation is exactly? So it's the bold signal, is the beta value from the correlation.
So you're talking about this curve?
It's accuracy, but there is some.
No, the neural data, it's activation. Yeah. What?
Yes.
So you're asking what is on the y-axis? Is that what you're asking?
Yes.
Yes.
Yeah. Okay. So this is an event-related design, and so we're looking at neural responses to stimuli like the ones that that I showed in this figure here. My apologies. And so we're looking at neural responses to the stimuli that are presented in this kind of a paradigm. So there's an adaptation phase, and so we can measure that independently. And then after several trials, and it's sort of variable how many trials of adaptation are shown. It could be 8, it could be 20. A stimulus is presented, and sometimes that's a deviant stimulus where it changes numerical value, it could change size, it could change brightness, or it could stay the same. And so we're measuring the neural activation to those stimuli that are either identical to the adaptation period or different in some degree. So that's what's being plotted on the y-axis, is the bold response to those stimuli.
I think Andrea's question was a bit more technical, and this comes because we're doing a similar experiment in the lab, not with numbers, but is asking exactly what is plotted in the y. Is it the bold response? Is it beta values, or is it t values? Is it? I guess that's his question.
Yes.
It's
Yeah.
a very specific question about the data. But
I
we can
see,
talk
yeah,
about that
no,
later also. In fact, we're going to
Right,
bother
yeah,
you.
but there are beta values from a regression that includes a factor for the adaptation period and includes factors for each of the conditions in the deviant stimuli.
Does that answer? I think that answers the question. But we're going to bother you for sure about this. There's other questions here also now.
Thank you.
Armando Machado from University of Aveiro.
Hello. Thank you. It was a lovely talk. I have done some research with pigeons on similar tasks. And one of the things that I was asking, how meaningful is it to assign a Weber fraction for a species, even though you call it a Weber fraction for number? The fact is that I don't know how meaningful are the comparisons, because we have done similar stuff as you have done, monkeys and children, but we have done with pigeons in serially presented stimuli. And when you don't present a stimulus simultaneously, when you present a stimulus simultaneously, you have all the confounds of spatial variables. When you present them serially, you have temporal variables, cumulative duration. But you retrieve different Weber fractions. So what's the Weber fraction of Armando, myself, on number? Well, depends on the task. So how do you compare tasks across species? Makes it even more difficult. And so I don't know. And let me just conclude by saying that the linearization of this, you start with a scalar property and, a nonlinear function, and apparently with humans, some kind of education linearizes the representation. Well, I'm not so sure, because when you go to those experiments like Dehaene has done with the Munduruku tribes and so on, there's very hardly you can see individual data. Very hardly, because initially what these people many times give you, as far as I understand, correct me if I'm wrong, is categorical stuff. When you average across people, and then you have a somewhat smooth function, and we have done the mental line experiments also with children, and it's not the case that they give you automatically linear functions. They give you categorical stuff, whereby a small numerosity is one side of the mental line or line, and all the other numerosities are on the right, on the other end. But if you average across subjects, lots of step functions, you have a smooth function. Oops, is the Weber fraction, how meaningful it is? And if you could elaborate how you conceive the linearization thing.
Yeah, so the first question is something that that we are also very interested in. And so one of the things that we did to get those Weber fractions, so the Weber fractions that I'm plotting for each species are a latent variable. And so we used subjects who had done a bunch of different tasks. And so we model a task effect and an individual subject effect. And we tried to come up with a Weber fraction that sort of simultaneously deals with those task effects and the individual subject variability in order to come up with something that's representative for the species. But it is very true that if you are doing a serial number task, that there are different interactions with the system. So for example, most animals are much worse at making sequential judgments than they are at making simultaneous ones. And partly that seems to be because a sustained attention is required in a sequential task in a way that it isn't in a simultaneous task. So there are task effects, but I think it is possible to try to account for them mathematically in a model that samples broadly enough across different tasks and across individuals in order to try to come to estimate for a species a Weber fraction factoring in all of those variables. And that's something we're working on. And so that's, we used a sort of Bayesian model in the paper that we're kind of working on for philosophical transactions that takes a stab at that. And certainly there's room for improvement and conversation about how to best account for that variability, but it's very interesting. And then what does that mean at a deeper level? So yes, I think, will that Weber fraction for that species predict a single trial of an animal's behavior, right? So no, but it will, in theory, predict the sort of aggregate responses of that species or clade, over time. And so it's something, it is interesting to think about what are all of the factors within an individual that contribute to the output of a numerical judgment. And so there'll be very general factors. There'll be, all the way through the processing pipeline, there'll be contributions of noise that will affect the final outcome. But I think it is ultimately a tractable problem to try to identify those, and that's sort of the goal of understanding the mechanism. And then the second question was about the aggregate performance versus individual performance. We fit individual data by including individual subjects effects in our analyses. And so we're careful to not make interpretations like the ones you were describing that would only emerge at the group level and would not emerge at the individual level. And so in the Munduruku, some of the behavior is a little bit different. So those are people who, people debate this, have only number words for like the first few quantities and have nothing else. And so there could be effects there. We see effects of culture on numerical sensitivity, and I assume that they would be seen there too, where you might get some kind of categorical effect based on their symbolic culture and the fact that it represents early numbers very precisely and later numbers not so much. You might see some effects of that riding on top of their estimation performance. But my sense, based on what we're seeing in monkeys, what we see in adult humans when we prevent them from counting, what we see in kids over many, many trials fitting individual functions, is that you will see these ratio effects of estimation. And it's not, you'll see kind of, the kind of pattern of ceiling effect plus, monotonically decreasing performance with numerical ratio as subjects are estimating these values. But there could be categorical effects riding on top of that.
I think we have another, thank you, Jessica. I think we have another question from João Peça.
Hi, Jessica. Thank you for the very nice talk. João Peça here from the University of Coimbra. First, I'd like to start apologizing of following such questions of, at the level of granularity. I'm afraid mine will be maybe a little bit naive. But 2 points I would like you to comment on. One of them is that for me, I don't know if it wasn't entirely clear if there are ethological behaviors that would predict the ability or the necessity for an animal to display a better performance in the ability to differentiate larger numbers from smaller numbers, and if that somehow with the species that you've looked at. So that would be one question. The second question is, are there any gender effects? Have you looked at this possibility? And if yes, in which species? Thank you.
So in terms of the kinds of natural behaviors that would put pressure on a species to develop finer sensitivity to number or sensitivity to larger quantities, there are lots. People haven't, I don't think the field has gotten to the point where they're able to link precise cognitive mechanisms with specific behaviors that we're seeing in the wild in a causal way. But there are sort of observations of the kinds of problems that animals solve that I think are good candidates for why an animal would need to do it. Some of them are foraging. And I think sometimes we humans think about foraging problems maybe too simplistically compared to what they might be like for an animal that has to survive out in the wild. And so foraging problems for animals, it's not like looking at two discrete patches and deciding which one has more and which one has less. It's sort of traveling over space and time and keeping mental track of how productive a particular area is in terms of the food that it could provide for you. And then also weighing in how many competitors from your own group are occupying that and depleting the resource. And so there's a range of sort of complicated problems that an animal could consider, and there's some evidence that they do consider at least those two factors, the amount of food and number of competitors for making foraging decisions. And then there's also interspecies competition, there's between group competition, and there are social problems that the animals need to solve. And it sort of depends on your group size, how large of a number of individuals you might have to consider. Certainly if you're a social insect or a fish, that number could be quite large. Maybe if you're a monogamous primate, that number wouldn't be large. So that it's not clear exactly in terms of the large number problem how that interacts with the animal's social behavior, but there are considerations that a lot of animals seem to make in terms of, you know, what's a good bet or what's a good next action based on the you know, the quantities of competitors or of allies that the animal is dealing with. And so there's some data from chimpanzees and from lions about their willingness to approach a fight between groups based on the number of allies that they have with them and the number of opponents that they have in the approaching group. So there's you know, there are social problems, there are foraging problems. They're probably more complicated than what we currently understand. But there's certainly a range of problems that could put pressure on a species to develop a finer Weber fraction. And then in terms of gender differences in species, so I have not seen that very much. You know, we don't necessarily have the sample sizes to ask that question in animals. I have looked at that in humans, at least in early childhood, for a range of mathematical tasks. And they're early in development, both behaviorally and in the brain. So we've done some whole brain analyses comparing young boys and girls on their neural processing profiles, including amplitudes and variability and sort of the shape of their time course. And we don't see gender differences early in development on any of the numerical or mathematical behaviors that we've looked at, or in the aspects of neural development that we've studied. And so in a lot of these basic processes, I don't predict that we'd see gender differences there. It seems like the pattern we see in humans is that gender differences in math, in formal math, and evolutionarily recent math is something that emerges socially and culturally. It starts at about age 8 or so, and it's been decreasing over time, over generations.
Okay, Jessica, I should stop here, but I can't help myself and ask a question also, and that is, it's related to your data. But why IPS? IPS would not, if you didn't know this and you just asked, where should an area for number be? Well, whether it's number or not is a different question, or whatever, numeracy, whatever you want to say. I would never say IPS. I know it is, but you know, it's a visual motor area. Does it have to do with the digits and stuff? But that doesn't make, I don't think that squares with the animal literature. So do you have any idea why that would be the case?
Yeah, and so that's really interesting, a really interesting question, and I do think about that question. So I think one reason might be, you know, and this is the blind watchmaker phenomena, right? It's not like evolution puts things, you know, where they logically go. It just sort of grabs what it can, and so one nice feature of parietal cortex for number representation is that it integrates information from a lot of different regions. It's multimodal, and so we know that number representation can be auditory, it can be tactile, it can be visual. You need to be somewhere that you know integrates a lot of information. So that's, you know, that's not a blind watchmaker thing. That would be sort of optimal. But the blind watchmaker thing might be related to the fact that neurons in parietal cortex like to accumulate information over time. And so there's a lot of decision-making processes, even some of the motor representations that all have that kind of functional profile of accumulation over time and space. And so, you know, it might be that number is an object-oriented phenomena. In order to represent it, you have to integrate information over space and time about discrete objects. And it might be that both the functional profile of neurons in that region and the way information feeds through the system, that's where, you know, that kind of representation could emerge. And so then this again gets back to what I was talking about earlier, which is about the constraints, right? So you have to kind of think about the evolutionary, the developmental constraints, the information processing constraints and the hardware and how it's set up. And you, of course, you know this, but it's that joint satisfaction of all of those things that will land you, I think, in a place like parietal cortex for this behavior.
Great. Thank you very much. It's a great talk, and again, thank you for accepting our invitation.
Yeah, thanks, George.
So we're going to move to the last part of the of our conference, and I want to ask Vice Rector for Research, Professor Cláudia Cavadas, to join me here. But before she gives last words and she closes the session, I just want to thank obviously all of you for being here, and the speakers, many of the speakers for fantastic talks. There's also a couple of people that I want to thank. Danielle has been the invisible or not so invisible person organizing this conference, so thank you, Danielle. And also a bit invisible, Miguel, the IT service has been fantastic, and thank you again. Thank you so much for this. And finally, after the fantastic closing speech of Cláudia, there is a reception at the cloisters, and so you should join us there. But first, and most importantly, Cláudia.
Okay, good afternoon, everyone, and thank you for the invitation to be here. So when Jorge told me and sent me the invitation to be here for this closing session, I thought, should I be the Vice Rector? Then I realized that in fact the program was the Vice Rector. Should I be the researcher? Should I be the friend? Or the professor anyway? So let me see if I can do like a very good speech, enthusiastic. So, first of all, when I saw the title of this 3 conference, I thought, very, very interesting. I want to go. Then he invited me to come as Vice Rector. I said, not like as a researcher. But I'm not joking. The people that know me, that for sure I felt like that. So, but first of all, I'd like to acknowledge the team that put this together. Jorge, Óscar, and all the team together. So this is really very interesting. So the organization, of course, the speakers. So I was attending by YouTube. So I was in other meetings. So I was attending. So it was really outstanding communications and outstanding piece of work. So it was wow. And of course, all the researchers and the colleagues that are contributing to this field in the world, and of course in the University of Coimbra. So, when we see this event, so in fact this has all the ingredients that a university needs. So excellence science, basic science, crossing different fields, collaboration between people, between researchers in with top-level researchers, internationalization, and of course, again, I will repeat, interdisciplinarity, so even it's in the title. So thank you again to put this together. But if we look for, so when I saw this, the speakers that were here, I said, so we have people that I know already for a long time, like João Peça, Patrícia Maciel. So I said, we have CNC here, Center for Neuroscience, a research center with 30 years. Of course, we have ProAction Lab, we have CNAIC. So then I was looking for the 38 research centers that we have at the University of Coimbra, and I was picking. So who should be also here collaborating and listening? And I did not did like a very profound study, but I was at least 7 or 6 research centers could be here to try to push this area of brain, crossing between psychology and neuroscience. So it's really very, very interesting, and thank you, Jorge, to push this area in the different perspectives. So I think it's really very interesting. So then, of course, we do need this kind of events to attract more people for these research areas. So this is to attract more talent. So the ProAction Lab, led by Jorge, has been very successfully to attract talent. So we hope that University of Coimbra can have the power to attract and keep them and nurturing their career. So this is really very, very important. This is a challenge for a vice director. This is really very difficult. But in fact, if you look for any university in Portugal, we have 2 levels, and these 2 levels are very difficult to combine. So maybe I will not be very, so maybe it's not interesting for young students and young research that are starting, but in fact we have 2 levels of faculties. We are here at the faculty of pharmacy. No, it's my faculty of psychology, and we have the research centers. So the research centers are where the science is built. Of course, then we'll come to the faculty. So these 2 levels sometimes are difficult to bind and to combine. So since I've been working since the beginning of my career at the Center for Neuroscience, that is an interdisciplinary research center, I always felt this should be the way to work. Putting together researchers, having the scientific questions, and it doesn't matter where are you from. This is my point of view, maybe not very well perceived, but this is the important thing, as we have to do very good science, and where is the box, where is the, doesn't matter. So this is my point of view. So and I'm very proud to say that at the University of Coimbra we are doing a lot of efforts to keep and to push the research in other areas, or at least to help the researchers to be here at the University of Coimbra and to attract more. So we have different initiatives that started not long time ago, like trying to attract more ERCs, like Jorge, or to keep them, the ones that are here. Marie Curie, so we call it ERC at UC, Marie Curie at UC, so not very innovative, but the numbers are increasing. It's not very, we cannot see it very well, but we are seeing that in the paper. Gender at UC, this one I'm very, very proud, because you are pushing the gender in terms of including gender in the research. So we have to fill the gap. And so when we say gender in research is during from all the steps in research, including the content. So this is a very innovative initiative that we have at the University of Coimbra. You have even being so the Europe look at us, so they are fantastic. So this is, I think it's really a very interesting. And now we have a new project. So all of this, we have some of investment from the university, some of other investment from European money. So we have one that now we are pushing the career development for researchers. So this one will be very interesting, that we should be like University of Coimbra wants to be like an excellent place to keep researchers and to give them career development. So this is a new research project that starts have the kickoff today. So of course another one, it's doctoral schools. So we have to push the early researchers to giving them transferable skills to be excellent researchers, but also being prepared to the world. So this is so we are pushing. It's a big university. We say the 38 research center, 1,500 around researchers, so 25,000 students, and one vice rector for research. So it's a lot of barriers, but I think you are slowly keeping and have this kind of events, and people like you are here, and the collaborators. I think step by step we will reach another level. And thank you for your support in research. Keep doing good research. Keep nurturing the friendship and collaboration with good people, and And thank you. Thank you, George, and thank you, Óscar.
Thank you very much. We'll certainly keep on doing this. In fact, we will do this again in 2 months. There's going to be another one of these conferences in March. Another one. We'll keep you updated. Thank you very much again. And that's it. That's the end. Thank you.